So, hello everyone. My name is Noémie Coulon. I'm a postdoctoral researcher at the University of Montpellier and a L'Oréal UNESCO for Women in Science fellow. When we think about the decline of sharks, skates, and rays, we usually think about overfishing. But another threat is emerging, more silent and often overlooked: climate change. And today I'm going to show you how this threat could affect something fundamental, their ability to reproduce. But before going further, I would like to thank all of you for the tremendous work done on sharks, skates, and rays biology. This review is built on your contributions. I screened more than 100 studies covering about 88 species. So really, thank you, and please give yourself a round of applause for this tremendous work. So, what does this collective work actually tell us about reproduction? To answer that, we first need to clarify what we mean by fertility. This process relies on 4 key stages, from gametogenesis to mating, eventually sperm storage, and postzygotic development. And at each step, environmental constraints can reduce the probability of successful reproduction. So, fertility is not a single process. It's a cascade of potential bottlenecks. At the gametogenesis stage, temperature plays a key role in endocrine regulation. It directly controls hormonal cycles in spermatogenesis and ovarian development. For example, in the small spotted cat shark, temperature alters spermatogonia, cells that give rise to spermatozoa in the testes. At the mating stage, many elasmobranchs only come together during specific seasonal aggregations. These aggregations are tightly linked to environmental cues such as temperature, prey availability, or habitat conditions. But climate change can disrupt these cues, shifting distribution, altering timing, or degrading key habitats. This may lead to mismatches between male and female. For example, in the angel shark, the absence of female during a breeding season in 2022 coincided with a major thermal anomaly in the Northeast Atlantic. At this stage, many elasmobranch females can store sperm for extended periods, from days to months, even years, allowing fertilization even when mating is not synchronous. However, sperm quality is highly temperature dependent. For example, in the bamboo shark, the sperm stored at room temperature shows reduced performance compared to 5 degrees Celsius. This suggests that warming could compromise the viability of stored sperm, reducing fertilization success. At the post-psychotic stage, climate change can directly affect embryo development and survival. In oviparous species, embryos develop externally and are directly exposed to environmental conditions. For example, in the small spotted cat shark, up to 90% of embryos die under end-of-century warming scenarios. In viviparous species, females can partially buffer environmental conditions, but temperature still affects gestation. For instance, in the Atlantic stingrays, 1 degree Celsius increase can shorten gestation by as much as 2 weeks. More generally, while moderate warming can accelerate development, extreme temperatures can severely impair survival and physiology. So, at this stage, climate change can directly reduce recruitment and population renewal. Some mechanisms may partially buffer these constraints. For example, in the small-toothed starfish, parthenogenesis has been documented in wild populations, allowing reproduction in the absence of mates, but producing offspring with reduced genetic diversity and very limited reproductive potential. Another example is embryonic diapause. In the Australian sharp-nosed shark, southern population exhibit a prolonged diapause and higher fecundity than the northern ones, suggesting a role in matching reproduction to environmental conditions. However, both mechanisms are likely sensitive to temperature, and their role under climate change remains poorly understood. Which raises a broader question: why it is so important to understand how temperature affects fertility in elasmobranchs. First, for sharks and rays, this is critical. They are an ancient and highly diverse lineage, but with very low reproductive rates, which makes them particularly vulnerable to environmental conditions. In fact, climate change is likely to amplify existing pressures, such as overfishing, further increasing their extinction risk. And beyond elasmobranchs, many of the processes we discussed, from gametogenesis to sperm storage, is shared across mammals, snakes, insects, and even birds. For example, the blue shark has evolved a placenta-like structure, a feature more associated with possum. So at the same time, their exceptional diversity of reproductive strategy makes them a powerful model to understand how climate change may affect fertility more broadly. Of course, these are just a few examples, and I'd be very happy to discuss this question further. And I invite you to explore the full review for a more comprehensive picture. And thank you for your attention.
Thank you very much. We got time for one question. Anyone? No? Okay. Thank you very much. Next up, we have Dr. Leigh Livne, sorry, with Reproductive Physiology of Dusky and Sandbar sharks in the Eastern Mediterranean, a mixed method study of movement health and gestation status.
Hi, everyone. As I was introduced, my name is Leigh Livne. I'm here representing the Morris Khan Marine Research Station, the University of Haifa in Israel. Thank you to the organizers for having me, and it's really nice to see a lot of friendly faces in the crowd. The BBC came to film our site for their Asia series, and I thought it was the best site and footage of our species, both Dusky and Sandbars. Although it's really a sin to mute Sir David Attenborough, I will keep it to ourselves. This is the Hadera aggregation on the Israeli coast. What you're seeing is a predictable winter presence of large Dusky and smaller sandbar sharks in very shallow water. Sharks arrive when the temperatures drop to around 23 degrees, around mid to late November. And the warm water effluent from the station is around 10 degrees higher at this time. This aggregation has been monitored since 2014, but sharks have been reported along the coast for decades. It is one of the only places in the Mediterranean where mature individuals can be repeatedly accessed and sampled. But the aim of my project is to understand the biological underpinning of this aggregation. It's correlated with warm water effluent from the station, but this is not the exact species-specific reason. So we structured the work around 4 questions. Is the aggregation reproductive in function? If so, are females arriving at a specific gestational stage? Do the gravid sharks return annually or on a longer cycle? And based on above, how does this all affect the management of the species? Also, this aggregation was largely sex segregated. Female Duskies adults and adult male sandbars, but also that pattern has begun to shift slightly with every season, with more female sandbars, lucky for me. So now my study includes them as well. Dusky sharks are slow growing, late maturity, we all know this. This combination places them amongst the most vulnerable to exploitation in the Mediterranean. And in the Med, they are classed as data deficient. There are very few reproductive studies, I believe only 2 in 1965 and 1 in the 90s, both from bycatch studies outside of this region, but it's accepted that they have a triennial reproductive cycle, so 2 years of gestation and 1 year of rest. Sandbars are much more common in the Med, and we know them much more, and it's very well established they have a biennial reproductive cycle. In a rapidly warming Eastern Mediterranean, it is understanding how and when these animals use these coastal habitats become more urgent. Oh, I'm also very proud to say that in Israel, the sharks and rays are fully protected. My approach combines 3 methods, but I also added later on 2 contextual pieces of evidence. The first is ultrasound to determine gravidity and the embryo size, complemented by plasma hormone analysis to assess the reproductive state, satellite telemetry to examine movement and habitat use. And for context, I added in later our long-term acoustic monitoring program that we use to assess multi-year presence. Last year, I started compiling more of the data as I got involved with the strandings and the necropsies from the sharks. And the goal is not to rely on any one single data set. It's important to say that we have very low recapture rate. It's an aggregation, not population. So we're looking for agreement across independent lines of evidence and to patch things together. You can play the video. From 2018 to 2025, 18 adult female Duskies were assessed on board. 17 were gravid and 1 was nulliparous. Embryo sizes were tightly clustered, around 30 centimeters, estimated stretch total length. The range was narrow and most individuals were in early gestation, with only 2 sharks classed as mid-gestation, which were also caught later in the season. We did not observe any late-term embryos across all years, and when scanning for follicle size, they were also very difficult to locate, and I wasn't surprised as the pups are in early stage, the follicles will be around 10 millimeters, very difficult to see. The nulliparous individual showed a thin uterine lining on the scan, and combined with her small size and the follicles that I very rarely saw, we were ready to mate in the upcoming season. For sandbar sharks, we saw a different pattern. We know that they have a biennial reproductive cycle. So when we caught her in March, I was not surprised to see that she was late stage, around 45, 50 centimeters pups, and this aligns with their known cycle. Endocrinology aligned also with these scan results. Estradiol and testosterone profiles are consistent with more uterine maintenance rather than ovulation or late-stage pregnancy. The nulliparous female showed elevated estradiol, suggesting a pre-ovulatory state. And for the rest of the data set, the profiles support that they are active gravid females in early stages. The elevated E2 in the late-stage gravid sandbar likely reflects the concurrent vitellogenesis that is preparing for the next cycle because they are biennial. Given the low recapture rates, it's difficult to resolve the full hormonal cycles, but combining endocrinology with ultrasound has proved to be very consistent so far. Moving on to movement. One of these gravid females from the scan earlier, our 97th shark, was tracked for 6 months and fitted with a new low-tech birth alert tag, an archival sat tag, also from low-tech, and acoustic tag, and a lot of jewelry went on that shark. She spent roughly 80% of her time in the epipelagic zone. in a narrow temperature range of 22 to 26 degrees. Her deepest dive was in September, in 240 meters, around 15 degrees Celsius, with deeper dives occurring at the hottest times of the year. But geolocation estimates from the tag were not reliable enough for spatial interpretation with huge error. We'll say that all the points were located within the longitudes of the Eastern Mediterranean basin. Combined with public reports, the sharks look to be moving away from Hadera at the borderline months, so at the start of the summer and the start of the winter. And then they moved deeper and offshore. The tag popped up in October when the water was 26 degrees at 45 meters depth. This is very interesting in terms of the reproductive cycling. It does match with all the other analysis that I'm putting together. Now moving on to the multi-year returns and the necropsies, the contextual evidence. We started acoustically tagging in 2018, and every year since around 2021, I've been secretly checking to see if there was some sort of 3-year return for the females. And in 2025, I did a full review of the data set and noticed that 4 of the Duskies that we tagged, 1 in 2018, came back in 2021, and 3 that we tagged in 2022 came back in 2025. So this is roughly a 3-year interval. Although we don't have scans back then, the study was started in 2021, the pattern is consistent with the triennial cycle, and since almost every, except for 1, female that I've scanned on board has been pregnant, I'm very gently assuming that these were also pregnant. The Sandbar males have always come back year on year, and we're getting more Sandbar females and seeing that they are too coming back year on year, which is really interesting. We also had a stranded Dusky this year in January with 7 embryos, all around 33 to 34 centimeters. This was sad, but exciting for me. I was finally able to get a total length with a girth estimate so that I could better estimate my stretch total length in my scans. This closely matches, in January, the same time of the scans in the water that I'm doing. All of them are around 30, 32 centimeters. Importantly, I was able to look at the follicle size at the time in January, and it was exactly at 10 millimeters, which is what I expect to see and what I'm seeing on the scans as well. Putting this together, Hadera functions as a gestational stage-specific aggregation, not a mating site. We don't see new scars. Oh dear. We don't see evidence of parturition. For the Dusky sharks, the aggregation is dominated by early to mid-stage gestation. The narrow embryo size distribution, agreement with necropsies, absent of late-term embryos and return intervals, when assessed against known studies, suggests that the gestation is approximately 18 to 20 months, before the only literature estimate for this species is 16 to 22. So this fits within, but it narrows it down. For Sandbar sharks, additional scans and tagging are needed to resolve the gestational habitat use. We're collaborating with many researchers in the area on a Nat Geo project for this specifically. We require a network approach to ID the parturition grounds so that we can understand how to further protect these species outside of our borders. Most importantly, I think Hadera, given the lack of recaptures, this mixed methods approach has been very helpful in starting to put the puzzle together. And I want to say thank you to everyone. Oh dear, Dr. Shainin and everyone.
Thank you very much. Next speaker is our first student talk, Ítalo Fernández-Cisternas. He's going to talk about valuing kelp as nesting grounds.
Thank you everyone to be here. Today I will talk about what is important to give a conservation value to kelp forests for underrepresented sharks with an example of cat sharks that we have in Chile. And this is because sharks don't perform parental care, so several species relied on nursery grounds to secure the development and survival of the offsprings. Several functional habitats serve as nursery areas for these species, like kelp forests for oviparous sharks and mangroves for viviparous sharks. However, despite the important role that this functional habitat has, most of the functional habitats around the world are already vanished. And some of them are losing their physical attribute complexity, that with that are also losing their functional role that they give to these associated species. So, however, some species have shown plasticity in the use of these nursery areas. So it is give us a kind of hope that some population will have some resilience in terms of the habitat degradation. So this is why it's so important to understand how dependent are these species for the physical complexity attributes of this functional habitat, and also how much these species use these nursery grounds. So today I will talk about this species in Chile that is a cat shark, a very small cat shark, oviparous shark, endemic from the Humboldt Current System. The special thing about this shark is depend completely on the kelp species of Lesonia trabeculata to lay the eggs, since there is no other substrate that have ever recorded to lay the eggs. And so you can see here, it's not just about the species, it's just about a microhabitat the species use for lay the eggs. That is exactly in this point between the stipes and when the fronts start. So we can imagine, since there is no parental care, female has to find the best habitat quality to lay the eggs. So this is why I asked two main questions. To what extent does the morphology of Lesonia trabeculata determine its use as an imposition substrate? And if this pattern is going to be consistent along a latitudinal gradient, or we will find some plasticity in the use of these substrates? So this is why we perform several different diving along the most of the distribution of this species and the Lesonia trabeculata. So we find 8 oviposition sites where every time that we find an egg-bearing kelp, we measure 3 important morphological attributes: the holdfast diameter, the number of stipes, and the total length of each kelp. We also perform transect. We also do the same morphological measure with non-bearing kelp individuals, and we also count adult individuals and juvenile individuals of kelp just to have a measure of the density of the forest. So what you see here, we have the 3 main different morphological aspects of the kelp forest. So you see the density of the kelp individual measured. So what you see here is egg-bearing kelp that is in orange tend to have wider holdfast diameter, more stipes numbers, tend to have larger total length than non-egg-bearing kelp bearing that is in gray. And this pattern was consistent across different latitudes and was independent of the size structure of the kelp population in each site. We're talking about the kelp forest configuration. We only found a negative relationship between the number of eggs per elk and the density of juvenile kelp forest. So that also means that it's not only the kelp forest has to be present in these areas, but also they have to show some complexity that the juveniles doesn't give to the shark for oviposition behavior. The other aspect that we want to know, understand is, okay, they rely on these specific attributes of the kelp forest, but we also want to understand how much these species use this oviposition site. And this is because what you see here in this work, there is possible to find fresh elk capsules along the year. So that means that the females lay eggs during the year. So we can think that these oviposition sites have an influence on the species distribution, especially for females that stay in there laying eggs more than males. This is something that I don't invent. This is the classical framework that is for other vertebrates like mammals and birds, and also sharks have, where females stay very close to these nursery grounds and are the males that the vector of the dispersion for the populations. So First, we see that these species relied on these very localized oviposition sites, and they use this non-random kelp morphology along a very long latitudinal gradient. So now what we did after that is just to perform this fine spatial scale study. So you can see here, this is an array of receivers. A receiver is, they were located in a very small array. You can see the 600 meters just. And you can see there is two patches of kelp forest, and both patches has a specific oviposition site, that is the triangle, don't you see? So we tagged 10 males, we tagged 10 females with this acoustic tagging for more than a year. So we were able to scope 2 reproduction seasons. An important thing, just very quickly about how this work, we tag these animals with this tagging, they generate a specific sound. So every time that it past through the receiver's range of detection, it will give us the direct time that this animal was detected. So we obtain this animal have very fidelity to just one specific receiver. So that is why we want to understand how these individuals are structured in the area. So we perform this network analysis with nodes or receivers at edge as connection between the receivers, and we did this fast graded algorithms just to detect cluster between animals. We designate each individual to their most favorite receiver. So that is why we calculate these clusters. And after that, we perform this home range just to know, to look visually how they use the area. And then with the fast graded algorithm, we were able to detect 2 different clusters. And you can see here for the number that females were more segregated than males. They are exactly the same cluster, movement cluster population. But the most important thing is what we have to see. You remember that triangulars are the oviposition sites? So for females, the group that is found in the south, they never visited the northern oviposition sites, and vice versa with the females from the north, from the oviposition side. And this is what difference for males, that they were able to be detected in both oviposition receivers. So we think this is kind of social behavior that female has. They tend to avoid males, and also there might be some competitions for the oviposition site in the very small area. Compared to the males, they just move between the areas trying to breed with females. So in summary, what we can see here is this species not only use this specific microhabitat in a specific species, that is Lawsonia trabeculata, in a specific site of the Lawsonia trabeculata, but also they use these very localized breeding sites with a specific, they have to master a specific habitat characteristic to lay the eggs. And finally, we find this very fine scale fidelity to these oviposition sites. And so what this means, that this species is highly vulnerable to habitat loss and degradation. And what is important here in Chile, because right now we have this huge problem with overharvesting. Fishers are extracting the larger kelp, so that means there is only the kelp is disappearing, but it's also is getting more complex. So this can affect completely the input of the offsprings for the next generation of local population. So we have to integrate all these parameters to management and conservation of kelp forest. Thank you so much to be here. I'm always glad to thank to all the international collaborations, Savior Seas, and all my friends from my lab that without them this work have been even possible. So this is part of the project. You can find it in Savior Seas. I'm right now looking for a postdoc, so if you are interested to collaborate or know more about this project, I'm gonna be here. Thank you so much.
That was excellent timing on the second. No time for question, unfortunately. Next up, we have Youri, Ivann, Mvomo, Minko. Evidence for the use of southern Gabonese coastal waters as nursery grounds for Sphyrna, lewini, and Carcharhinus limbatus.
Good morning, everybody. I am Youri. I represent you this work about nursery sharks in Gabon. So in coastline waters in Gabon, we have different fisheries activity. And artisanal fishery represents the mainly components of this activity of fisheries. In 2010, artisanal fishery accounting nearly 17% of national catch of Gabon. And in this artisanal fisheries, there are a locality in southern Gabon which represents nearly 70% of catch of sharks and ray. This catch of sharks and ray are mainly bycatch and represent 25% of total catch. And in this catch, we have two species. We represent the mainly species of sharks and rays catch. And what is interesting here, that these species are critical and dangerous for scalloped hammerhead and vulnerable for black-tip sharks. And also, there's two species using nursery as a life-seek strategy. So for us, our aim is to demonstrate the presence of a multi-specific sharks nursery for Sphyrna lewini, scalloped hammerheads, and black-tip sharks in Mayumba in southern Gabon. We have also different specific objectives, mainly access the importance of these species in these regions, also evaluate different criterion about nursery sharks in southern Gabon. Just show you the study area. We are in this region, Mayumba is here, and the fisheries, smallscale fisheries occur along the coastline south in Gabon. This part of the littoral in Gabon in particular is composed of lagoons. Different for other sides of littoral Gabon, we can see we have a delta and estuary. To take our data between April 2022 and March 2023, we have record sharks and ray data. We also considering fishing efforts and use CPUA. We also taken ventral muscle tissue for how two species sharks, but we are also taken tissue for milk shark. This species is interesting for us because they are proxy species because they spend entire life in this littoral. So we have also take historical data fisheries. Is market 2088 is wrong, sorry, is an error. It was in 2015, 2014. This data, OSHEF data collected from April 2014 to March 2015. We also taken data in 2017 and 2018. This is historical data we have taken to take consideration how analysis. We also taken the different analysis about mainly body size and fisheries, stable isotope analysis also. And this, we use stable isotope analysis to prove criteria 2 about sharks nursery. For criteria 1, we use juvenile abundance is in your size. For that, we have used the same fishing gear. This is a bottom gill net. This is a principal fishing gear of artisanal fisheries in Gabon. So for criteria 3, For analysis, we have used seasonal interior CPU estimate with 20 GLMs with different predictions. So, what we can see in the slides, scalloped hammerhead and black-tip sharks are nearly caught only in those regions. We have a few catch of the species in other regions in Gabon, very few compared to Mayumba. And we have also seen that these two species represent the main catch of the regions. For scalloped hammerhead, we have nearly one third of the total catch. And we can see also mixed shark represent also a main catch of these regions. And we have seen different maturity stage of how two species, and we have seen the predominantly composed of immature individual. We have also seen neonates and juvenile, according to the period of the year. About body size, mean body size, we have seen there are significant difference concerning black-tip sharks according to the month of the years. No difference was observed, no significant difference was observed in scalloped hammerhead. So we use this critter with a stable isotope of carbon and nitrogens to compare the probability of niche overlap between how two species with mixed sharks, and how results showed this overlap of trophic of isotope niche is higher. So when we compare historical data and data taken during our study, we have seen their presence was always motions of two species in these regions. We have different interannual catch with black-tip sharks. So, we have shown scalloped hammerhead and black-tip shark were the main elasmobranch species landed by the local artisanal fisheries with concomitant occurrence. We have tried to demonstrate the three criteria to establish a nursery in these regions. It's very important, these results are very important for us because with the government of Gabon, we are trying to establish a sanctuary sharks in these regions. Basing of this data and other data, we want to explain that the area where artisanal fishing of Mayumba exists can be established in nursery because they represent for us a part importance for the conservation of these species. So when I thought we have a 20 regional shark conservation strategy with different partners, mainly national parks. So for us, the sanctuary are a main base for to be established. Thank you.
Thank you very much. No time for questions. The next speaker is Nicolas Tolan, second student, and on characterizing a black-tip reef shark nursery, multi-method insights from Malaysia.
Hello, welcome. My name is Nicolas Tolan. Very excited to be here to be sharing some of my doctoral research, primarily our methods of characterizing black-tip reef shark nursery in Malaysia. So, a little overview. Elasmobranch research in Malaysia. The South China Sea and the Malay Peninsula is a very understudied region for sharks and rays. In particular, the black-tip reef shark, Carcharhinus melanopterus, very few studies in the region, population connectivity, a little bit about the trophic ecology, and one small study about conservation planning in the marine parks. Large knowledge gaps still remain. Abundance within these marine parks, identifying nursery habitats, and ontogenetic residency patterns. So, Chagutang Bay, a very small but growing group of publications have centered on Chagutang Bay in Peninsular Malaysia. It's a small island gazetted as a marine park in 1995, and at this particular site, the Sea Turtle Research Unit at University of Malaysia Terengganu have been conducting conservation for over 30 years. Why study sharks at the Sea Turtle Sanctuary? Well, 30 plus years of conservation has led to limited development, human disturbance, really healthy coral reef fringe ecosystem, and a seasonal abundance of sea turtle hatchlings. And everybody knows sea turtle hatchlings are the chicken nuggets of the ocean. So this Chagutang was also designated as an important shark and ray area in Israel last year in 2024 for its potential as a shark nursery site. This is all led by evidence from student-led studies, including baseline assessments of abundance diversity, stable isotope analysis of the juvenile shark's trophic role, and their documentation of the exclusive predation during the nesting season of juvenile neonate sea turtle hatchlings. So let's explore the shark nursery criteria, because that was the main focus of my study, to characterize Chagutang Bay not only as a sea turtle sanctuary site, but as an important black-tip reef shark nursery site. So higher abundances, are there more juveniles in this area than others? We did some extensive abundance studies around the whole archipelago, and what we found is Chagutang really does have some high abundances. Also, we want to look into their site fidelity or residency. How long do these sharks stay once they're born as neonates within the bay? Is it days? Is it weeks? Is it months? And the third criteria, is this site used as a nursery site or a birthing area over multiple years? So our objectives and methods. Our first objective was, again, assessing the abundance of juvenile and adult sharks at Chagutang Bay particularly, after a very extensive assessment of the three large islands. So our methods were bruvs and drones, and then our second objective was evaluating the residency, the site fidelity of the neonate sharks, and we particularly used mark and recapture, traditional mark and recapture, and acoustic telemetry. So what's our evidence from bruv studies? So a quick overview. This was some of our methods. So we wanted to look at the spatial and temporal abundance. So we're looking at different reef zones. We have in the yellow the shoreline, the reef fringe. I divide it between a midreef and a forereef because that midreef is a little more lagoonal, where it gets really deep in that forereef. And we use bruv and drone surveys across these areas, bruv surveys and drone transects to try to understand the abundance of both adult and juveniles. And what we found here was that there are way more juveniles, of course, in the shoreline area, really restricted to this western shoreline side, while we saw more adult sharks, significantly more adult sharks in the deeper forereef areas. And this is with bruv surveys. What we found from our drone investigations was actually a bit complementary results for the juveniles. We saw a really high abundance of juveniles, CPU, within the shoreline, but we also saw high abundance of adult sharks in the shoreline as well. Luckily, we also didn't see any juveniles. So for both bruv and drone surveys, no juveniles were detected in the deeper zones. This next group of figures is actually my research assistant, Larry Zhang. He was tracking the sharks with the drone, so he'd centered the drone over the shark. And you can see in figure C, those are the swim tracks of the sharks. He put it into pretty much area usage, and then he also characterized the shoreline and then the midreef instead of my three areas. But he had some complementary results where in the reef flat, that shoreline area, we saw the most abundance of juveniles, but also some adult sharks. What about our evidence from integrated mark and recapture and passive acoustic telemetry? Well, all sharks that we caught were neonates. They're all under 62 centimeters. Our mean days at liberty for mark and recapture was about 56 days. And you can see we had our highest catch per unit effort in May and June. That actually coincides with peak sea turtle hatchling. No sharks tagged in 2023 were recaptured the next year, which to us means that they're kind of, once they're growing large enough, they're leaving the bay. And then 2 of our acoustically tagged sharks out of only 5 individuals were in the bay for more than 90 days. And you can see here, integrating our methods, integrating mark and recapture and our acoustic telemetry helped us extend our residency estimates a little bit. You can see our mark and recapture, days at liberty, so basically our first capture and our last recapture. And then our acoustic telemetry in orange is basically from when the sharks were tagged released and when we ended the study. Another important feature is our west receiver stations. We only had 2 receiver stations within the small bay. The west side had the majority of the detections, over 80%. And for that, for our juvenile sharks, kind of helps us understand that there is a specific habitat within the bay that they prefer. So I guess the main questions that we wanted to understand is, did we satisfy Hypo's criteria? So the higher abundance, yes, we saw that there was a lot higher abundance of juveniles than adult sharks, especially along the shoreline. From the drone evidence, we saw that adult sharks were also abundant within the shoreline. That just goes to show the differences in the methods between our BRUV detections, the BRUV being a single camera for about an hour out in the ocean, where the drones is an active transect and able to cover more area. We also saw there's some spatial preference. So unlike areas in the Pacific where you might have a large lagoon, that's a really important nursery site, it's a very spatially tight nursery habitat for these juvenile sharks along the coastline. Site fidelity or residency. So 96 days acoustic and 133 days mark and recapture. Those are our estimates for how long our residency periods were. Our integrating tagging extended those estimates. And then the third criteria, repeated use over time. So BRUVs, drones, mark and recapture, they all recorded neonate sharks in 2002, 2023, and 2024. However, we're always left with more questions. Particularly for me, is if these sharks are not staying for longer periods of time as neonates within inside the bay, where are they actually going? And what's their home range extent? So here's a little map of Redang Island, and Chagutang is a very remote bay at the top. However, there are sightings of juvenile neonate sharks within some of these larger bays. And our questions are what's the home range extent? Are these sharks moving to other bays and moving back? And another really big question, movement patterns of connectivity between islands. So there's a bit of an archipelago or marine parks, Brahenti and Lantana, Redang, they're all semi-protected as a marine park area. And we want to understand, we want to have a better understanding of juveniles and adult sharks, what are their movement and connectivity between these islands. So thank you. You can see my email. We have our first publication out, the Spatiotemporal Patterns and Associations of Black-tip Reef Shark in a Shallow Tropical Bay. Please check it out. Any questions?
Any questions? Yeah, one question. We got a mic over there. So,
hi, great talk. I was curious about for the BRUVs, when you were saying you found the differences between the number of juveniles between BRUV and drone surveys. Do you think that could be like competition for the bait camera, like with adults going for the camera or being?
So actually what we found is the juvenile sharks are way more attracted to the bait and would stay around the camera for longer periods of time. This is also found in other research as well. The adult sharks that we found in these BRUVs, we never had one adult shark come and actually interact with the bait. Most of the time they're just passing the camera by. And that's what we particularly found with the BRUVs. With the drones, the reason why I think we found a difference in the abundance of sharks in the shoreline with drones is mainly the drone just has a larger area it can scan. It's about 300 meters, the transect, and it's within 5 minutes. And we always find in the morning at these areas there's a lot of adult and juvenile shark activity in the morning. And one of the hypotheses is this release of sea turtle hatchlings, a constant food source for them along that habitat. Any other questions?
That's all we've
That's
got
all
now.
time?
We gotta
Okay,
mute now.
sorry.
Identifying critical nursery habitat for juvenile blacktip reef shark in the Gulf of Thailand.
Hey everyone, my name is Kirsty and I'm here representing New Haven Reef Conservation Program and talking about the work that we've been doing to assess our blacktip reef shark populations here. So one of the first things is understanding the area that we're actually working in. So the area that we have is a small tourist destination that's called Koh Tao. Quite popular. We have a lot of people coming here specifically to do some form of in-water activities. And we've been working on the population of black tips there going back to 2011. And it's something we're continuing to do. So our goal with this was really to identify the nursery habitats as well as assess the trends that we're seeing over time. And also incorporate a way that we could work with our students and interns to integrate them into data collection as well as data analysis. So the reasons that this is important are we know that nursery habitats are critical to the long-term survival of shark populations. It's somewhere that they are being repeatedly used over time. And we know that globally, all of these habitats are under threat. Now, some of the issues we're facing within Thailand are, number one, it's a data poor region. There are a very small handful of us that actually do any research on sharks and specifically populations of sharks within Thai waters. And those of us that do generally work on multiple species because there's only a few of us. So our goal was really to help us understand more about our native populations and what we can do moving forward to help understand their movements as well as understand how we can better protect them. So the methods that we were using for this are standardized surveys. So we were using snorkel surveys combined with towing a GPS in the earlier stages and recording on the GPS every time a shark was encountered, as well as standard data of the life stage, the behavior, and any interesting other information that we come across with. And in later years, we've also switched over to using RUVs. And the reason we're using RUVs rather than BRUVs is simply because the area that these guys are usually found is a very big tourist spot. So we didn't want to run the risk of introducing bait and then having a much bigger problem on our hands. So usually our RUVs are deployed for about an hour, and we do this periodically throughout the week, sometimes in the mornings and sometimes in the evenings, and that gives us a good shift in terms of the data that we're able to collect. And the reason we chose this area is, number one, historically we've always seen sharks in this region, or in this part of the island itself. It's a shallow water depth. Average depth is about 4 meters. So it's perfect to be able to study this area. It's very sheltered from the currents, especially during pupping season, and we don't really have shark predators in the area either. So everything that we've found so far signals that this is a very important area in terms of nursery habitat. So what we're seeing is throughout the course of the year, so this study is looking from February to August of 2015 to 2025. And what we found that March and April have the highest abundance of sharks being observed in the area. Now, part of that is in the March, we have a lot of pregnant females coming into the bay, indicating that this is in the lead up to the pupping season. And in the end of March and beginning of April, we then start to see an increase in the number of neonates, indicating again that this is the time that these guys are giving birth. Now, because we've got data going back to 2011, this is a consistent pattern that we're seeing year on year, again indicating this as a nursery habitat. But one of the concerning things we're now starting to see is the decline in our number of observations. So I mentioned we go back to 2011, but 2015, we had about 1,100 observations of sharks during our study surveys. In 2019, that number declined slightly. In 2023, it increased slightly, and in 2025, the observations within this bay dropped down to 440 observations. And this isn't because the effort was any less, because actually we conducted more surveys in 2025 than we did in 2015. So the actual effort has increased, but numbers are not providing this. Now, one of the other things we have noticed with 2025 is a movement of some of the individuals. So neonates, we're now starting to see more in a neighboring bay. And if we combine our survey on neonates from that bay along with our normal study site, the number of observations that we found jumps up to about 850, 860. So again, if we're combining these two places, we are more in line with what we've previously recorded, but again, it is signaling this movement and a strong resilience and sensitivity. And again, just to reiterate that point, if we look at the bottom left, we can see year on year from 2015 to 2025 comparisons, the numbers of these different life stages has been quite significantly different. And in these cases, again, that drop in the neonates is a really big cause for concern with the research that we're doing and trying to understand why that's actually happening. One of the other things that was quite noticeable throughout all of our surveys was the distinct lack of males. So there were few, but they were nowhere near the amount that we were expecting to see. And the RUVs have proven to be quite useful, but we don't really see neonates on the RUVs. Or if we do, it's very few and far between. So combining that RUV with the snorkel survey has actually been very important to identify the different individuals and the observations themselves. So one of the biggest cause for concerns that we are starting to look at right now is understanding some of the risks and the threats that are facing these animals. So some of these threats include things like our ghost nets, hook and lines, we have entanglement, and we have bycatch. And although fishing is not particularly prevalent in Thai waters, we know that it does still happen, and we still encounter things with fishermen collecting these animals without understanding the true value of them. And we also have a lot of issues, which is the reason why we think there's been a decline with increased tourism. So Koh Tao itself gets upwards of 500,000 visitors a year, and of those 500,000, it's about 90% of these people that get into the water for some kind of snorkel-based or dive-based activity. And Koh Tao is actually the best place in Thailand to get in the water with black-tip reef sharks, and also to encounter sea turtles. So you can imagine the level of tourism that is now being pushed onto this particular bay. Land infrastructure is another big problem as they're trying to deal with all of the tourism increase, and of course, marine debris. But all of this research has been quite instrumental in terms of, again, a data poor region. So the work that we've been doing has also helped us to designate Koh Tao as an important shark and ray area, both through our black-tip reef shark research and our whale shark research. But this isn't enough to really protect what looks to be a declining population. So our next idea is to then really start to understand why this decline is happening. And we're hopefully moving into using acoustic arrays to understand whether the individuals have just been pushed off into the deeper water because of the increases in tourism, and more specifically look at, as we've seen them arriving in different bays, whether that's then starting to be a more prominent pattern. So we still have 2026 to conclude the rest of our survey data for this time, and then we'll jump into trying to sort out the acoustic arrays. But it's definitely something that is incredibly interesting, and a huge part of the economic value of Shark Bay resides within these sharks. So again, it's something that's very important in terms of determining why this decline is happening. And if anyone has any questions, I would be happy to take them.
Any questions?
Hi, Kirsty. Sorry, I'm a bit at the back of the room. I've got a quick question. Have you looked at any temperature data together with those declines? I don't know if you had the heat waves in the Gulf of Thailand as well in 2016 and 2024, but I feel like that's such a big overlap. Do you have any plans in looking into that?
That's one of the next things we do want to look at, because at least in terms of the snorkel-based side of things, there's been times where we've got in the water to do the surveys, and we're acknowledging that it's incredibly warm for us. So I can only imagine the issue that's causing for the sharks. So it's definitely something we're going to be looking forward to moving in.
Yeah, epic. Thank you.
Okay. Next speaker, we've got Steve Gerber on Beyond Movement, revealing reproductive Israel on Africa's southernmost coral reef.
All right, welcome everyone. My name is Steve Gerber. I'm a PhD student at Nelson Mandela University in South Africa. And I have to admit, I applied for this, but I was still in the middle of data analysis, and I've gone down so many rabbit holes in the meantime that the whole narrative has shifted a bit from the original title to Beyond Nursery, revealing high school ISRAs on Africa's southernmost coral reefs. Sorry for the trick there. The coral reefs that I'm referring to are encompassed by the Isimangoliso Marine Protected Area. It's South Africa's largest coastal MPA. It's adjacent to Mozambique, and it's a multi-use MPA, so you've got different zonations with different regulations in it. The whole area is considered an ISRA, partially because it contains vulnerable species. There's the ragged-toothed sharks that come up seasonally to enjoy the warm water during their gestation period. And there's some occasional aggregations of rays. But overall, it's considered a movement corridor connecting the southern areas like Utukela Banks and Aliwal Shoal, which are super productive, with the more tropical areas like Maputo and Wazaruto National Park in Mozambique. And that's kind of what I want to contest here, because I claim there's a lot more going on than just sharks passing through. I've been deploying about 400 BRUFs over 2 years, looking at the classic MPA zonation effect, submarine canyons in my case as well. And what I noticed very early on is all these little guys. I want to call them toddler sharks, because otherwise everybody's got that song stuck again in their head for the rest of the day. And yeah, but if you're thinking like me originally, ooh, nursery, it's not quite what it looks like, apparently. So let's get into it. Starting with the white-spotted wedgefish, critically endangered ray species. And you can see here all the detections that I managed to measure in size. So there's the, on the x-axis, the seasonality, on the y-axis, the size measurements that I took. And the first thing that struck me, obviously already watching the videos, is, juvenile, ooh, neonates. Not super abundant, but they seem to be present throughout the year, but not enough to suggest a core nursery area, basically. Then I started looking at all the females and all the males, and where are the males? I mean, there's these three. And that was it. The rest was all females, massively female biased. And they're also not sexually mature yet. You see those dashed lines is where males and females respectively reach sexual maturity. So we also got really big females around year-round, except for when it comes to that time, and then they're just kind of gone. Now we actually do know the bigger picture of things, because down south in Utukela Banks during summer there has been pregnant female, and there are neonates down there. So it seems like a cohort of the population does reproduce in Utukela Banks. The fact that there's so many females around the rest of the year may indicate, and the female bias obviously, that this is a resting area either before mating or after pupping or both. And the fact that there's about 45% of all detections were subadults and juveniles. So it may indicate that it's kind of an area that they go to after their neonatal stage, kind of like high school basically. Now, the species can, at least in theory, maintain its whole life cycle in South African waters, and that's vital. I'll get to why in a bit. Now, I literally had this pattern repeat for quite a lot of species. I'm just going to show you a few more, starting with tiger sharks. Again, we have some neonates. Again, the vast majority is subadults, barely any adult ones. And again, the seasonality is quite striking, with significantly more detections during winter compared to summer. They start showing up in these southern areas during summer. Given the fact that it's mostly subadults, this probably doesn't have to do with reproduction, but just feeding areas opening up with increasing water temperatures. And then also the gray reef shark, more tropical species. Again, some neonates, mostly teenagers though, about 70% are larger juveniles and subadults. And again, strong seasonality. Here's the opposite pattern though. It's a more warm water species, and they seem to be a lot more abundant during summer, and then come winter, they might be heading a bit more into Mozambique again. I do want to mention the scalloped hammerheads as well, critically endangered. This is the smallest individual and the largest individual I got. This is too big to be a neonate, and this is too small to be sexually mature. So again, it's all juveniles and subadults. However, I don't think BRUFs are very ideal for hammerheads, because 400 BRUFs and 18 detections is not a lot. And we know that they're not uncommon in the area. The thing is, though, if we look at the fact that 16 detections happen in winter, only 2 in summer, we can still infer quite some seasonality. And in fact, it adds up with a great picture, which is exactly the same as for the wedgefish. It's a well-established fact that Utukela Banks is a pupping ground, a nursery ground. And it seems, again, like Isimangoliso is at least seasonally kind of this high school function of hosting a lot of subadults and juveniles. But again, I think we're missing part of the picture because BRUFs are not ideal for hammerheads. I also want to mention common black tips and silver tips, because I didn't get them too often on my BRUFs, but I do have hard evidence that it's mostly juveniles and subadults around. The thing is, this isn't scientific footage. This is me having my GoPro while freediving, so it's on a scientific basis of trust me, bro. But still, just to indicate, like, there's a lot more going on, and this is mostly BRUF-based, and we've already heard earlier this morning, there's obviously its own biases. Now, coming to the discussion, the first thing I kind of want to coin here is this winter residence hypothesis, basically. East Mongoliso appears to be the southernmost limit for a lot of these subtropical species during winter. And it may therefore kind of serve as a pole position because, you know, I mean in life, but in biology especially, you snooze, you lose. So if you have feeding grounds or reproductive events happening, you want to be one of the first ones to get there, right? So it makes sense for you to kind of wait at the limit of your temperature threshold until temperatures rise and you can go, assuming competition, prey availability, and everything is acceptable levels. The main thing I want to highlight here, though, is this high school concept. I call it high school because it's very snappy for representation. What I obviously mean is multi-species subadult developmental habitats. It's a bit of a mouthful, though. And yeah, East Mongoliso hosts mostly juveniles and subadults, it seems. And it's generally known that young of the year don't stick around the nursery area for more than a year. They very often start leaving after a while. And it seems like many of them in the region then move into East Mongoliso to kind of grow up for a couple of years. The area is also known as a teleos nursery, which means great prey availability, especially for smaller, less experienced predators, right? Now, why specifically East Mongoliso has this importance? I think it's largely because of its transitional nature. I've hinted at it already. It's largely because of the environment. It's between bioregions and climatic realms. And many species are near the temperature thresholds, which obviously becomes very interesting with climate change as we're expecting the southward expansion of a lot of these populations. So especially with the seasonal differences, they might slowly disappear. The main factor that makes it so important, though, is in terms of the transitional nature in terms of jurisdiction, because while this area is still within the population ranges of all these species, it is just on the other side of the border, and that is crucial, because we're all struggling, obviously, with conservation and protection. Africa is no different, but if you compare the situation to Mozambique, Tanzania, and Madagascar, South Africa is still doing really well. And if push comes to shove and these populations collapse, functionally at least, further up north, I mean, the genetic bottleneck is going to be horrifying, but at least it seems like these populations can be maintained or maintain themselves within South African waters, given that all the life stages are acknowledged properly and the areas that they require are protected accordingly. And this brings me to the take-home message. Now, I've said East Mongoliso so often, maybe just put that aside, put away the species and everything, and I kind of want to finish up on a thought experiment. If you have a species that reaches sexual maturity at about 7 years, what we're mostly talking about in research as well as conservation is the adults and then the neonates, maybe young of the year, the smaller juveniles. And then we talk about recruitment, but recruitment isn't the juveniles. Recruitment is how many will start reproducing again, and that leaves us with, in this example, a 6-year period that we're very often not really considering. We're only talking about nurseries and then where the adults are going. If you consider the ecological, the spatial ecology of these, neonates have ridiculously high predation pressure, obviously, and limited diet. If you compare that to the adults, low predation, diverse prey, but a lot of their movement is going to be governed by reproduction, mating, giving birth, or the recovery thereafter. But if you have this middle ground here, every year they're having a better size refuge, they're diversifying their diets, and I've described it here as platonic lifestyle, basically, because they don't have to bother about sex yet. And that's my take-home message. If you're like me on the nerdier side of things all your life, you probably don't want to remember your high school years too vividly either. But I urge you, don't forget about the teenagers, because it's a vital section of their life that the species needs for survival as well. And if there was a high school ISRA definition, I'm sure East Mongoliso would fit it, but I bet there's more high schools around the world as well. Perfect.
Next speaker, we've got Bradley Heyman with nutritional resource partitioning in South Australia.
Cool. Well, I just wanted to start today's presentation just by acknowledging how much of an absolute pleasure it is to be able to share some of my honours research on the international stage today. And just with that, to be able to thank OCS as well for providing me with some funding so that I can actually be here today. So yeah, we're going to start today by talking about animal niches. And an animal's niche is just the job or role that an animal will play within its ecosystem and incorporates all of its core resources that it needs to survive. For the purposes of today's talk, I want to talk specifically about dietary niches. So, in regards to dietary niches, the way that we quantify them is we have 2 core niche metrics, the first of which is our niche width, which is just the richness or breadth of resources consumed, and we can have a wide niche width with a lot of resources, or a more narrow niche width with less resources being consumed. From this, we can start to understand the feeding strategy of our animals, as a wide niche width is generally indicative of a generalist predator, and a narrow niche width indicative of a specialist predator. And again, from this, we can actually start to have a look at the possible ecological vulnerabilities of our animals as well, with some research looking into how ecological specialization can make animals more vulnerable to change. Our second niche metric that we use is this idea of niche overlap, whereby it's just the amount of resources being used by the same animals. So we can have a higher degree of niche overlap, or we can have a lower degree of niche overlap. So the methods that I use within my study, I have two. First of all, we have stable isotopes, which allow us to have a look at habitat through the carbon values that we get, as well as relative trophic positioning due to the nitrogen. One of the areas where isotopes struggle, though, is in terms of its taxonomic resolution. So let's just be hypothetical and say that these animals that I've put up on screen have the exact same isotopic values as one another. So we aren't able to discern what prey these are based on their isotopes alone. But if you compare a crustacean to a teleos, they're obviously very different in terms of their tissue and their composition, and they're going to be what we consider isotopically similar, but nutritionally distinct, which is where our second method comes in, and that's this idea of fatty acid analysis, whereby all animals, including ourselves, are only able to synthesize a certain amount of fatty acids naturally before we have to get the rest from our diets. So with an understanding of the fatty acids of our prey, so let's say our gummy shark eats our crab there, we can then actually track that through their fatty acid composition and have a look at what they're eating just based on that makeup. We can also start to have a look at other factors such as water temperature and habitat type. Moving on to what we already kind of know about resource partitioning in Southern Australia, it's pretty much summed up by this plot behind me. And within this, in South Australia, we have about 80 species of elasmobranchs. But in this model here, they've been put into 3 broad categories. So what we can start to infer from this is that these animals would have the exact same resource use, they'd have complete trophic overlap and occupy the exact same trophic position. But I kind of want to point out that we have 3 morphologically similar and commercially viable teleos species that have been broken down into the species level. So it starts to give us this idea that maybe that's not really the case. When we start having a look at global knowledge as well, we have a look at WA, Shark Bay. We can see that across these 13 species, they are carving out their own niche space to be able to coexist with one another. And again, with a study from South Africa, whereby these 6 species do have an overlap occurring, but they're still able to have their own kind of resources so they can live with one another. So again, kind of shows us that what we know so far might not be the whole truth and might be a bit of a generalization, which is where my study comes in. So I sampled 13 chondrichthyan species across Southern Australia, 9 of which I'm going to include in my talk today. And very important to note that I just used muscle for the purposes of my study. We're going to jump into some of the results, and I'm just going to try and pull out some of the bits that I find the most interesting personally, and I'm going to start with our Port Jackson sharks here. So if we have a look at the isotopic plot, plot A, we can see that there's quite a bit of spread in terms of their carbon value, but in terms of their nutritional niche, they're a little bit more narrow, which means they're a little bit more specialized. So what we can start to kind of infer from this is that these animals might be feeding on fewer prey types, but across a variety of habitats, giving them that spread in carbon values. Moving on to our Gulf wobbegongs and our southern eagle rays here, what I want to highlight with them is isotopically, these animals don't have any kind of overlap, but in terms of the nutritional niche, the overlap is a lot more. So again, what we can start to kind of find from this is that these animals might be feeding on similar prey types, giving them that nutritional overlap, but feeding either in different habitats or at different trophic levels, separating them isotopically. The final example I want to bring up is for our southern saw sharks, whereby they're kind of the inverse relationship of what I've just said. So nutritionally, they are not overlapping at all, but isotopically they are. So they might be feeding on different prey types, but either in similar habitats or at similar levels in their food web. Moving on to our box plots here, these are basically just going to allow us to have a look at what animals we're going to consider generalists and which ones we're going to consider specialists. So with our isotopic plot, we can consider these animals generalized and these ones our specialists. When we move on to our nutritional niche, however, we can see this is a little bit different because we're going to have a few less generalist predators this time, and we're going to have a few more specialists, namely our southern fiddler rays and our Port Jackson sharks, where isotopically they are generalists, but nutritionally they're specialized. So kind of moving on now, for anyone that doesn't know, in South Australia, we've been having a bit of a hard time with a harmful algal bloom that's been causing a lot of marine mortalities. So what I've done is I've taken the isotopic niche width and the nutritional niche width, and then I've scaled them by the number of mortalities that we have seen just through like iNaturalist. And I know I've been harping on about them, but I want to talk about our Port Jackson sharks and our southern fiddler rays again, whereby they are facing some of the most amount of mortalities. And this may be due to their nutritional specialization. I do also want to highlight that our elephant fish and our southern soil sharks are quite specialized in terms of their diet, but the area where they were sampled from wasn't as heavily hit by the algae bloom, but there is potential that they may also be vulnerable due to those kind of ecosystem changes. So kind of moving through to our conclusion now, what we've done is we've identified generalist and specialist predators, and really interestingly, this can differ in terms of their isotopic and their nutritional niche. We've also started to have a look at the overlap present with our generalist predators overlapping heavily with our specialist predators. And again, kind of bringing this back to the idea that our specialist predators may be more vulnerable to ecosystem change as we go forward. So a few things that I would just kind of like to drive home and drill into a little bit is we have found that unlike that model that I showed earlier, these animals are effectively partitioning resources to facilitate coexistence. We are also finding that a species can have a differing isotopic and nutritional niche in order to kind of partition those resources with one another. We're also seeing that nutritionally specialized animals may be more susceptible to change in the event of ecosystem or environmental change itself. So just with that, I would like to take a second to acknowledge everyone that helped me collect my data through my study, so everyone from the SEG lab, and again, I would like to thank OCS for providing me with some student support so that I could be here today and talk to all of you. Thank you.
We got time for one question. Any questions? Okay, so we've now got two.
All right. So today I'll be talking about temperature, oxygen, offspring size, and population growth rate across sharks. I am a PhD student with Dr. Nick Belvey at Simon Fraser University. Here we go. So as many of us are aware, at least a third of shark ray and chimera species are threatened with extinction, and at least 12.9% in the gray box in the middle are data deficient. So there's not enough information to properly assess them at this time. So what I'm interested in is trying to find correlates of extinction risk that will help us learn more about these species and evaluate their extinction risk. So one thing that we can look at is population growth rate. So many of us will think of population growth rate as changes in individuals over time, but what you can also do is calculate what's called Rmax, which is the maximum per capita population growth rate, and this is realized at very low population numbers where there's no density dependence. And this has also been used for fishing exploitation reference points, like maximum sustainable yield and other limits to reduce overharvest or unsustainable fishing practices. And this is also informative of recovery potential, where high Rmax species would have a better recovery potential after a depletion event, and also could indicate their sensitivity to extinction. But unfortunately, we only have enough data to estimate about 15% of shark ray and chimera Rmax values. So just like we were looking for a correlate of extinction risk with Rmax, now we're looking for correlates of Rmax in other things. So if we look here at our 72 shark species in our data set, you can see that there is a general negative trend. As you get bigger, your Rmax typically goes down. And this is because larger animals are usually in the slower life history category because they're big, grow slow, mature late, and often live a long time. That's compared to the smaller species that have a faster life history, where they grow fast to a small size, mature early, and have a shorter lifespan. But you can see that there's a lot of possible variation in this space and a lot of different combinations of size and population growth rate. So what I need to do now is help find other things to help explain Rmax other than just maximum size. And to highlight what this difference, this variance means, I'm going to highlight a few species for you. So our high Rmax species is going to be the Atlantic sharp-nosed shark, and then our medium Rmax species is the leopard shark, and our low Rmax species is the North Pacific spiny dogfish. And these are all within maximum sizes of like 1 meter to 2 meters, so they're roughly the same. So then if we simulate a recovery scenario, so we're going to start these species off at 100 individuals and see how they recover back up to an 1,000 individual number. We can see first that our Atlantic sharp-nosed shark has one of the highest Rmax values in our data set. And to make more sense for everybody else, I've calculated what's called population doubling time, or TD. And so this species actually has a doubling time that's less than 1 year. So that means every year it can more than double in size, which is very productive. And because of that, they can recover to 1,000 individuals in about 5 years. And then looking at the medium Rmax species, our leopard shark, the doubling time is longer at 2.5 years. So for this population to recover, it would take almost 25 years. And then our low Rmax species, the spiny dogfish, the doubling time is actually 10 years. So for this species to get all the way to 1,000, it would take 75 years, and I didn't want to make a plot that goes that far, but it's a long time. So because these things happen at the same size, I'm trying to find other correlates other than size, so we can look at temperature. So lower temperatures are usually associated with slower life histories, and warmer temperatures are typically associated with faster life histories. And for our two extreme species here, across the range of the Atlantic sharp-nosed shark, the average temperature is about 14 degrees Celsius. Then our North Pacific spiny dogfish is much cooler, and across its distribution, it hangs out around 3 degrees Celsius. So that's one avenue that we can look at. But we know that temperature and maximum size aren't exactly independent. This is where we start drawing our causal relationships. So we'll start by saying that temperature has a positive effect on Rmax, as we've expected. And then we'll say that we have a negative effect of maximum mass on Rmax as well, as we've seen in the first scatter plot. But we know that temperature has a tendency to reduce maximum size. So we'll say that there's a negative effect of temperature on maximum mass. And then we also can consider depth, as we know that deeper water species typically also have a slower life history and lower Rmax. So we can say that there's a negative relationship between depth and Rmax. But we also know, looking at a species, if it is a higher temperature species, it would be in a shallower depth. So this would be a negative relationship between temperature and depth. And then we can assume that gill size is like a metabolic activity proxy. And we know that bigger species will have more metabolic activity. So then bigger species would need bigger gills to accommodate that extra activity. And we also know that deeper species will typically have smaller gills, so there's a negative relationship there. And then because bigger gills would be associated with the shallow and large species, there would be an overall negative relationship to Rmax. And then recently we've been looking at offspring mass as another correlate, because sharks, if they're bigger, they typically make bigger babies, so there's a positive relationship there. And then if you're making bigger babies, you're probably making fewer of them, so there's a negative relationship from offspring mass to Rmax. And typically what you would do if you identified these as important, you would just group them all together in a temperature, or in a model that says Rmax is a function of all of these things added together, and just report your results. And this would be a good way to get a good model fit and sponge up all the variation, but unfortunately it would probably introduce a lot of biases and skew your results when reporting the effects of these things on population growth rate. So we have to keep these backdoor pathways in mind when we're building our models. So for example, if we want to know the effect of temperature on population growth rate, we would just have a model that is Rmax is a function of temperature, because we have nothing influencing temperature, and everything else is downstream. So everything else is kind of just noise at this point. So we ran a multi-level Bayesian analysis, which is fancy, but here we have our 72 shark species as the dots. The bigger the dot, the bigger the maximum size. We can see our model best fit as the line, and we can see that it is a positive relationship. And then the posterior distribution is the inset here, and it's almost completely to the right of the 0 line, so we're pretty confident that this positive relationship exists. So temperature does increase population growth rate. But here, if we wanted to look at the effective gill size, or oxygen uptake capacity, we need to also incorporate maximum size and depth, because we know that those influence gills, and then gills might influence population growth rate. So we include all 3 in our model. But when reporting, we're only going to report the one we're interested about. So here we can see that the effect of gill size on population growth rate is negative by the red line there. And the posterior distribution is mostly to the left, so it is confidently negative. And then again, looking at offspring mass, because we know maximum mass influences offspring mass, we're going to include both of those in our model. And then you can see that there's a strong negative relationship here. So the bigger your offspring, the lower your population growth rate. And again, the posterior is almost completely to the left of 0, so we're very confident that this negative relationship exists. And then looking forward, I'm looking to explore other correlates of population growth rate to add to these models, and also expand on the amount of RMAX estimates we have for sharks using informed imputation models that have been recently developed, as well as maybe developing schedules for reassessing species that have already been assessed to kind of quickly capture status changes, because if you're more sensitive, you might change status more quickly than others. Thank you.
We got time for one super quick question. No? Going once, twice. Okay. Next speaker is Kim Bassos Ho, Geographic Variation in Life History Trait in the White-Spotted Eagle Ray.
I got my own timer here, make sure I stay on track. So I just want to thank the host committee and the wonderful country of Sri Lanka for being amazing hosts, and I look forward to sharing our research on this species here. Well, actually, a group of species, the spotted eagle rays. So let's see. Yes. I want to thank my co-authors specifically from around the world. We have folks from Florida, Allison, France, Atlantine, from the Galapagos, Diana and Daniel, East Coast of Florida, Matta Jamian, and West Coast of Florida, Kristen. All research I'm presenting has been collected under permits and with the Galapagos National Permits and IACUC approvals. So a little background on the species I'm going to talk about. The Aetobatidae is 5 species globally. I'm going to be talking about 2 of them today. All are of conservation concern. They're listed as endangered, or a couple of them are threatened IUCN list. They have declining population trends, and it's because they have a preference for coastal habitats, and they're caught in fisheries and some targeted fisheries, and also are susceptible to pollution in these areas. So some of the questions we're asking is, the 2 species I'm going to talk about, are there geographic variations in their life history, and how does this relate to the habitats that they live in and food resources? A little bit of background, I'm not going to go into all the numbers here, but there are published studies on both species. Aetobatus nari is an Atlantic basin species. Aetobatus ocellatus is a Pacific basin species. A lot of the data has come from animals that have come up either in targeted fisheries or bycatch in the Atlantic. Some of these studies with the female and male sizes listed here. In the Pacific basin, they've come from some of the bather protection nets that they've put up, and some bycatch. So our one study down in the left was the one where there was actually live capture-recapture. So most of the data has come from, as I mentioned, targeted fisheries. The areas that I'm going to be talking about come from Bermuda, and that's in the Atlantic basin, about 1,000 kilometers off the coast. Florida, both coasts, east and west coast of Florida, and then the Galapagos Islands. And we're going to be looking, Galapagos Islands is the Aetobatus ocellatus. So just to put it in perspective, we're way over here in Sri Lanka, and most of this stuff is taking place over in the other part of the world. I think because this species, a lot of people ask me, is how do we catch these large pelagic rays? Some of these rays are over 2 meters. This is a sped up version from a drone of how we use a net with a net comes off the back, we circle the ray, it's in a net compass. Once that ray is in that compass, it swims around a little bit. So in Florida, we have this lovely specialized boat that allows us to do that. And the animals swim around for a little bit. And then to get a 2-meter ray up onto the boat or floating, we have our guys swim out and they do what's called a net taco. So they grab the lead lines and they'll pull it up to the float lines and the ray will be in like a net taco. And we're able to pull the ray up. That big floating pen is it's a game changer for being able to access and work up these larger pelagic rays. And this is where I'm going to ask you to just speed it up a little bit. Eventually we'll get that net over to we have a crane, we can pull it up, we can weigh these rays, get the things, we swing them on board. We have a tank on board that has oxygen, the animals kept in very healthy condition on board, and then later, same thing, taken off the boat and then safely released. So it allows us to do all our procedures there. So that's Florida. In Bermuda, oops, that picture came up first. Let's see. Well, the stuff in the back, it got buried by the video. But anyways, so the rays are sited also. Bermuda is slightly different in that it has a background of, it's like limestone. It's an old volcanic island situation. And anyways, the rays are used a slightly smaller net. But what I want to mention is if you look at the way that the hula is made, that's made out of PVC piping in a circle. So you can get creative about ways to handle these larger rays. That animal's actually wearing a video CAT cam camera on it as part of our research. In the Galapagos Islands, we go into these shallow lagoons using a panga boat, and you're looking and you can either cast net the rays, there's a little shark going by, or once the net's deployed, a half moon around the lava rock, and you can get the animals into a smaller area. So they're brought on board either with the cast net or with the net conference that you can be hand scooped out of the net. So once we're on board, our methods are we're doing all our sampling, morphometrics, we're weighing them, we're checking them for pit tags. We take a variety of tissue and blood samples. We do our acoustic surgery. These guys go into tonic immobility. We're able to do that. And for a few select rays, we've been able to do satellite tagging. And we lately have been able to use our portable ultrasound and do field ultrasound. So that floating pen, we can put them upside down. They go in tonic immobility. This is a big female that had 4 pups. You can see right there. Some of our results. So looking at the 4 locations, in Florida, we've sampled over 700 rays. We've had 38 recaptures now. So I'm going to be talking about the recaptures that are greater than 90 days at liberty. So in each of these locations. In the Galapagos Islands, we're at 231, and we just late the last field season, we had 5 more recaptures, which is great. So 23 recaptures since 2019. In Bermuda, there's been fewer recaptures, but we're working on that. So I do want to plug from the West Coast thing. So John Swenson's going to be giving a talk on Thursday if you want to see how important it is to collect those genetic samples. They can be used to estimate population. He's using a close kin mark recapture technique, looking at kinship and such. So visit his talk. So when we look and compare across the different locations, so remember Ocellatus is the Pacific Basin Galapagos species. The Florida rays get bigger. So following the color scheme on our disc width to weight length ratios, we find that, where's the pointer? Is this the pointer? I don't know. So the pink line, you can see the Galapagos rays, as they get bigger, tend to be a little bit lighter for their thing. And the other thing we're finding is our largest rays are females across both areas. Like the bigger rays get females. In fact, this picture down below is a larger female in the Galapagos with a younger, well, I was going to say a male chasing her, but he's about half her size, and he had claspers that were extended beyond his thing. As far as their growth, what we're finding is that the Galapagos grows slower. The von Bertalanffy growth curves for both females and males, we get a much higher K rate for the females and males from the west coast of Florida. We didn't have enough sample size to do it for Bermuda and the east coast, but where we've had enough recaptures and the numbers are over in the end. So with 10 females of recaptures on the west coast of Florida, K of 0.38. West coast of Florida, males with 17 recaptures, we've had a 0.26, and then in the Galapagos, we only had enough females to do it with 11 females, and we got almost half that, like at 0.13. Time at liberty is the two graphs over there, and you can see how the Galapagos rays definitely are growing slower. So the ocellatus is a slower growing ray. So in conclusion, why is this happening? Why do Florida rays go larger and faster? We think probably in Florida we have higher predation risk. There's a lot of sharks off our coasts. 8%, almost 9% of our sharks have, our rays show some sign of shark bite on them. We also have very productive shallow estuaries. Like you can go out and find all kinds of mollusk prey. Somebody in a previous talk was talking about the rays, and they do target these these bigger rays. The bigger they get, they can access these prey items. They can crack open these big horse conchs and these whelks, so to get a really productive diet. In the Galapagos, it's not quite as productive of that from the mollusks. The things you see are a lot smaller. But one of the things that we did notice is that looking at the historic literature, ocellatus in Australia do also grow big. They have gotten those there. And it may be that in coastal regions, that's why we consider that in coastal regions with estuaries and stuff, these rays have the ability to get bigger. It might be a predation risk thing. So this is stuff we want to dive a little deeper into. Another conclusion that's come out of our research is these long-term tagging studies are super valuable. So pit tag your animals, that potential to recapture them and get that growth rate is amazing. Oops. Is that the end? And lastly, I just want to say using drones and tools such as that to measure disc width is something that we're investigating. So I just want to thank our many supporters and funders and all the volunteers and student interns that have helped. Thank you.
Thank you very much. Next speaker is Rachel Eichitson with Rays Anatomy, which I think is a great pun, from morphological traits to ecological function.
All right. Good afternoon, everyone. My name is Rachel Eichitson, and I am a PhD student at Simon Fraser University in Vancouver, Canada. And today we are going to be exploring how we get from morphological traits to ecological function using rays. In the current biodiversity crisis, sharks, rays, and chimeras are threatened with an elevated risk of extinction. And while this alone is a problem, our understanding of the consequences of this potential biodiversity loss is limited, partially due to data deficiencies. We don't fully know what roles sharks and rays have, so we can't predict we cannot predict how ecosystems will respond to species loss. A potential solution to this problem is trait-based ecology, which links individual organism traits, often morphological traits, to ecological function, or the role species play in their ecosystem and environment. This, however, requires some additional steps to get from morphology to ecological function to bridge the gap. One option is to understand the relationship between morphology and gills to connect us to metabolism, which then underpins life history, which then shapes species function due to energy and resource allocation. So now that we understand the logic behind the morphology to ecological function pathway, where do we start? One example is in sharks with a trait called caudal fin aspect ratio, or CFAR, which is the surface area of the caudal fin, or tail, divided by the height. And CFAR varies from pretty low in species such as the nurse shark to quite high in species such as the longfin mako. Previous work has connected CFAR to mean gill slit height, where sharks with more athletic tails have proportionally larger gill slits, connecting morphology to metabolism since this captures oxygen in through the gills and oxygen out through swimming or activity. So, this builds the first part of our morphology to ecological function pathway. We've also learned that CFAR is related to swimming speeds and geographic range size, where species with higher CFAR tails have larger ranges, which are all indicators of dispersal or movement ability. So, we can see how we get from the morphological trait, caudal fin aspect ratio, to the ecological function of dispersal. Much of the data for these analyses have come from the same source, a very handy book called Sharks of the World, whose morphological accurate illustrations contain both gills and tails. So with all this work done on sharks, naturally we turn to the sister taxa, the rays. There are about 650 species of ray, and the relationship between morphology and metabolism is largely unexplored for these weird and wonderful fish. Fortunately, similar to Sharks of the World, there is a nifty book called Rays of the World, which contains illustrations for over 600 species. However, there is one small issue if you are trying to connect morphology to gill slit height, and that is there are no drawings of the ventral side or underside in Rays of the World. To further complicate things, rays also have 3 swimming modes, unlike sharks, which only have 1. There are caudal propulsion species, such as sawfishes and wedgefishes and electric rays, which use their caudal fins or tails to swim. There are also pectoral undulators or wigglers, such as stingrays and skates. And finally, there are pectoral oscillators or flappers, such as eagle rays and mobulid rays. So given this context, where do we turn to for morphological data? The answer is museums, which house hundreds, if not thousands, of specimens. Importantly, museum specimens also have dorsal and ventral sides, which makes it possible to measure both fins and gills. So our quest for morphological data began in 2023 with a visit to the California Academy of Sciences in San Francisco, followed by the BD Biodiversity Museum, the South African Institute for Aquatic Biodiversity, and most recently, Europe in October 2025, where we visited the Natural History Museum, the National Museum of Natural History, and the Museum of Nature. At each museum, we scour the ray collection and take measurements of 18 traits in person and take photos of some features that we analyze later. We've measured 768 rays from a total of 198 species. So, with all of this data, we look back at these 3 swimming styles and expect that the morphological trait most closely related to gill slit height will vary based on the propulsion method. So, for our caudal propulsion species, we expect that this will be caudal fin aspect ratio, or CFAR, and we expect that this will be most closely related to gill slit height. For a pectoral undulating or wiggling species, we hypothesize that the length to apex, or the distance from the tip of the snout to the widest point, will be best correlated to gills. We also split our undulators into two groups based on morphology, those with trunk-like tails, such as skates and round rays, and those with whip-like tails, such as stingrays and whip rays. Finally, for our pectoral oscillators or flappers, we expect that the ratio of the disc length to width, or the length divided by the width, will be correlated to gill size For the sake of time today, we are only going to be looking at just our caudal propulsion and pectoral undulating species. So in order to actually analyze all of this data, we first exclude immature individuals because at this time we are not interested in ontogenetic shifts in metabolism, and so we are left with 454 mature individuals from 164 species. We then divvy up our data set based on the 3 swimming modes, and then analyze our data using Bayesian multi-level models, accounting for phylogeny since we expect that traits evolved along the tree. Getting to our results, we'll start with our caudal propulsion model, which has 166 individuals from 56 species. On the x-axis, we have caudal fin aspect ratio, or CFAR, and on the y, we have average gill slit height. Here, each point is an individual, and each line is a random draw from our model. The median draw line is emphasized, and overall, we see a positive relationship between caudal fin aspect ratio and mean gill slit height. And that's great because that is what we predicted and wanted to see. Moving on to our pectoral undulators with trunk-like tails. This model has 173 individuals from 62 species, and now our x-axis is that length to apex. And again, we see a positive relationship between that length to apex and gill slit height. For our pectoral undulators with whip-like tails, our model has 71 individuals from 37 species, and this model is not a very good fit. That is basically a flat line, so there is no relationship between the length to apex and gill slit height for these species. So we went back to the drawing board, and when we look at just our pectoral undulators with whip-like tails and the morphological disparity between species in this group, we notice a pretty big difference between the proportions of the disc or pectoral fins. So if we take disc length and then flip it, and then we highlight disc width, we can really see that difference in variation, where the length divided by the width varies from about 0.5 in butterfly rays to 1.2 in some stingrays. So now, if we look at that disc ratio versus gill slit height, voila, we see a beautiful positive relationship between that disc ratio and gill slit height. Going back to our morphology to ecological function pathway, our results define the morphology to gills relationship, which underpins metabolism, which then unlocks the rest of this pathway. We can continue building the bridges on this pathway by comparing the morphological traits we've identified to measures of swimming speed and genetic connectivity. We can also use the relationship between fin morphology and gills that we have identified to make predictions for species that we haven't measured using rays of the world, as well as species that are newly described that are likely lacking data. Given the current biodiversity crisis, it is difficult to justify the targeted sampling for rare or threatened species, yet data deficiencies severely hinder conservation and management efforts. We can overcome this problem with easily measurable traits that connect species form to function, and complement costly, time-intensive sampling techniques. By developing meaningful indices of ecological function, we can impute and predict missing traits for all 1,265 plus sharks and rays. A huge thank you to everyone that has been involved in this project so far, especially the museum staff that are so accommodating and let us just rip around their collections. Super appreciate everything they do for us, as well as all of the research assistants that have helped in various stages of this project. And thank you all for listening, and I'm happy to take questions if there's time.
No time for question, unfortunately. You can find Rachel after this session. We've got one more talk before we break for lunch, and that is Emmanuel Midinudeva, Demography and Exploitation of Daisy Stingray in Benin, West Africa.
Yeah, thank you for having me. So I'm going to talk about a small stingray that we have in West Africa. And this morning, Leigh Guido has introduced well my talk of this I will say afternoon. So the context is that we all are keen to protect those species on this image. They are the one on shark on CITES list. But the challenge is, where do we have the smallest one? The smallest inhabiting only a certain area in the world. So there came the study. So they are really diverse in terms of the form, and the previous presentation also well introduced that the challenge that we do not have data. So without data, how are we going to protect them? Coming from a region we always call lacking data, so there's a need to look into it. So do we have enough data to think about the conservation? A challenge. So, and how can we successfully enforce CITES regulation for those species that are part of the daily fishing? So here is the species of interest, Fortitrigon margarita. It's quite difficult to differentiate it with the congenial margaritella. but through genetic analysis, we were able to, discover what it is really. So we have it only in West Africa, from Senegal down to Angola, and this species, endangered species in the region. So the study area is the whole coast of my country, Benin, in West Africa, and we cover all the coastline. So basically, data collection, we were measuring the disc width and the disc length of the species. That is the basic data we collected, even though much more data has been collected behind. So in terms of analysis, we look at the sexual maturity, length at first maturity, and all those things that can be looked for basic life history of the species. And the analysis was run through the R packages, Trophish R, ggplot, ggfishplot, and other. So what are the results? We work from 2024 to 2025 because these are preliminary results. And opportunistically, we sample because we could not sample everything that we had at hand. Wherever you go at any time, you find Fortigrigon margarita anytime you want. So we dealt with 248 specimens. 52% were female and 48 male. In terms of the first maturity size, so for the male are mature around 20.7 centimeters, and the females are mature around 26.6 centimeters. Now, let's look at the growth parameter in the region. This is specific to my country, Benin, not to Nigeria, not to Ghana, not to Senegal, but specific to my country. We use a genetic iteration-based elephant to assess and to evaluate what could be the mean size that both female and male could reach in terms of disc width. And we have 69 is around 70 centimeters. But recently I was able to measure 75 centimeters as disc width for a female, which means that the current data we are having could need to be reassessed. Anyway, but we can also see that in terms of lifespan, averagely the species can live up to 33 years old, can be 33-year-olds. That could be checked through aging of reading the vertebra of the species to find exactly what is the maximum age that it can reach. In terms of the mortality, so we have a mortality of, and this is natural mortality, not that based on the fishery, but natural one, is around 0.3. Now, let's look at the exploitation parameters. The red line, that we are seeing is, and the point we are seeing is the size that has been considered, and those are the majorly fish size in the country. So, here we are. Most of the size that are fished are around 23, 28, 33 centimeters. And the age corresponding are around 3 and 6 year or 7-year-olds. These are really old one because we have seen recently very young one, neonate in the sampling. Looking at the exploitation parameters, so the total mortality is around 1.2, and the fishing-induced mortality is 0.9. So we have mortality that is really high for the species. The structure of the population and the recruitment we can see that the largest size, the size that are the most present in the species and the population is around 25 centimeters, and the recruitment occur mainly around March, April. So in terms of the current status of the species in the region, as we can see, we have a mortality induced by the fishery higher than the natural mortality, and that can cause a decline of the species in the region. But we expect that and suggest to the fish managers that they should keep, they are fishing moderately. I mean, we say that this is moderately overexploited because we are not fully in the red part of this graph. If we were completely in the middle side, so it means that we are in big trouble. But at this level, we are cautioning the managers that they should carefully maintain or reduce the effort of fishing. Unfortunately, this is a bycatch species, so we don't know how it's going to happen. But we suppose that they fish 40% of the spawning stock biomass that allow the species to revive, to replenish in the region. So thank you. I'm really grateful to Save Our Sea Foundation, Mental Trust, and the Fishery Institute in Benin for supporting this work. Thank you.
We're now going to break for lunch.
I'm just waiting for the last few people to come in. I'm going to start things a little earlier just because I want to give a little more context to this whole science communication session. It's relatively new to the whole symposium circuit, especially with people presenting research and stuff like that. So I'm sure everyone is very intrigued and curious about what it's about, but I kind of just want to introduce it a little bit better and explain why it's so important that science communication and people being able to present on ways that they're going about communicating their science is so important in the research world. Okay, so firstly, I'd like to welcome everyone to the science communication session. My name is Jade Schultz. I'm the Global Communications Manager for Save Our Seas Foundation. So in 2022, the first science communication session was introduced at the Sharks International Conference in Valencia by Paul Cox and the Shark Trust team. It was important to Save Our Seas Foundation that we continue to build on the success of that success this year. As we all know, in just a few decades, human activity has pushed a third of shark and ray populations to the brink of extinction. This is a failure of humanity. However, it is not a failure of science. The research in this room and at this conference is extraordinary. The data exists, the knowledge exists, the solutions in many cases exist. The problem is, science has a powerful opponent. Every audience we communicate with arrives with their own story about sharks that has already been written. Whether it's a fear shaped by decades of sensationalist media or generations of cultural and economic relationships with these animals, or not knowing anything about sharks at all. Our job is to understand that story before we try to change it. We are not just communicating science, we are meeting people exactly where they are and sharing our knowledge in a language that they understand. This session is about bridging the gap between science and the general public. How do we move our knowledge out of this echo chamber? From community outreach to documentary films, from storytelling to visualizing data, we are going to explore every tool available to us because the stakes demand nothing less. So let's kick this session off with Jasmine, who will be presenting on tips and tricks for leading impactful media campaigns for complex conservation topics.
Hi everyone, thanks so much for being here. So I'm Jazz, I'm the Media and Communications Manager at the Mantis Trust. And today I'm going to share some practical tips and tricks on how to turn complex conservation topics into media campaigns that actually engage people, even if you have limited time, budget, and resources. So it's very easy to get caught up in the bubble of science and academia and assume that a global audience understands what we do, but the reality is the majority do not. So how do you take something that's highly technical and turn it into something that people actually care about and act on? So that's the role of the media campaign, to act as the bridge between us, the scientific community, and the public. So I'm going to walk you through this using a campaign that we led called Save the Mantas. It's a global education and advocacy campaign focused on improving protections for manta and devil rays. So the challenge, and I'm sure this is something that a lot of you feel as well when you're trying to communicate your science, you've got a very complex message, you've got a global audience, and you have limited resources and time. So we were working towards a proposal to uplist manta and devil rays to CITES Appendix 1 at COP20, which is the highest level of international trade protection. So it's a very technical policy process that most people are never going to naturally engage with. But public awareness and pressure can still play a really, really important role in supporting these decisions. So that essentially led to one key question. What's the simplest but most compelling version of this story that we can tell that stays true to the science while giving people a clear action to take? So from that process, we developed a simple framework, 5 steps that can be applied to almost any conservation media campaign or even to communicating your own research. So whether you work in media already or you've never done communications before, these steps can hopefully be adapted to whatever resources that you have available. So the first step is to simplify. So essentially, you want to be defining your key message and then stripping away all of the jargon. So instead of a technical policy sentence that we have at the top here, we created something simple and public-friendly, Save the Mantas. This doesn't mean removing the science, it simply means translating it into something that is accessible while still being backed by evidence. Essentially, if a 12-year-old can't understand it, it won't spread. So the second step is making something that is shareable. So with the Save the Mantas campaign, we created a clear campaign identity. We had a hashtag, a logo, a landing page, and visual assets that people could easily download and share on their own social media. We also created campaign t-shirts and encouraged supporters to wear them, take photos, and share them alongside the campaign messages on their socials, whilst encouraging them to sign a petition to be presented to delegates at CITES COP20. So what this did was it turned our audience into active participants, generating content and reach without increasing our internal workload. Now, I just wanted to share a few examples of some other organizations that have used simple, catchy campaign names to communicate very complex conservation issues. So you can see just a few words, a simple hashtag, that then you can use to provide supporting evidence for the campaign. So step 3 is defining your audience and your call to action. So you always want to ask yourself 3 things. Firstly, who are you trying to reach? Where and how will you reach them? And what do you want them to do? So at the Manta Trust, we defined our 4 key audiences and aligned them with the most appropriate channels and actions. So different audiences require different approaches. For example, high net worth individuals or major donors or corporates may be encouraged to donate. Members of the public may be encouraged to sign a petition to present to these delegates. And divers or scientists can help to amplify the message within their own communities. So the key is to think really strategically about your audience, channels, and actions so that you can streamline your efforts at the end of the day. Now, the 4th step is collaboration, and this is where campaigns really scale. Instead of doing everything yourself, you bring in external power, such as journalists, public figures, and partner organizations. So we work with public figures and influencers with over 2 million followers combined, who wore our t-shirts and shared campaign messages with their audiences, which massively extended our reach. Then when it comes to pitching to the press or collaborating with journalists, you basically need a really strong hook before you pitch. So ask yourself, why does your story matter? Why is it globally relevant? And what is your call to action? Now, if you can answer those very clearly, your story becomes much more pitchable. Now, please bear in mind there are journalists here at Sharks International, so don't be afraid to talk to them. At the end of the day, they need your stories. And we also collaborated with NGOs, media groups, and research organizations working towards similar goals. So we co-created content, which allowed us to share the workload, and ran joint petitions. So rather than building everything ourselves, we used external networks to scale impact. So here are some of our campaign advocates wearing the t-shirt and amplifying the message in our campaign video.
Manta and devil rays are some of the most majestic and intelligent creatures in our ocean.
But they're under serious threat, and time is running out.
All 9 known species are now listed as threatened on the IUCN Red List.
In some places, their populations have declined by more than 90%.
They're caught as bycatch or directly targeted for their meat and gill plates.
But for manta and devil rays, that kind of sustainability doesn't exist.
If we don't change how we manage and protect them, they could disappear forever.
This November, governments will meet at CITES to decide their future.
We're calling for manta and devil rays to be uplisted to CITES Appendix 1.
This would ban all international commercial trade in these species.
It would help shut down meat and gill markets, giving rays a fighting chance.
This decision could be a turning point, but only if the world is watching.
Every image joins a mural at the CITES conference, a global call to save the manta.
Sorry, I'm not sure why there was some audio issues there, but hopefully you could see the captions okay. So our final step is using the tools available to you. We relied heavily on free tools like Canva, DaVinci Resolve, and stock libraries such as Coggia and Ocean Planet. You don't have to be an incredible photographer or videographer to create these campaigns. There's so many archives of footage available to scientists, researchers, and conservationists. And we prioritized partnerships and collaborations over paid advertising. Finally, one very important thing, we built a clear timeline and we stuck to it. Campaigns move super quickly, so having structure behind the scenes is essential. So what did this look like in practice? We reached over 3 million views on socials, generated more than 25,000 petition signatures, and secured international media coverage. We also raised funding and engaged thousands of people through outreach and education. And all of this was achieved with a very small campaign budget, without paid ads or external media teams. But the most important outcome was real-world conservation impact. This campaign contributed to the successful uplisting of manta and devil rays to Appendix 1 under CITES, the highest level of international trade protection. And it showed that digital engagement, when done well, can support real conservation outcomes while staying grounded in the strong science. So to summarize, if we want people to care about conservation, we need to meet them where they are, make it accessible, and give them a clear way to engage. Thank you.
Does anyone have any questions for Jasmine? We have like 30 seconds. Okay, cool. Next. Oh, one question? Wait. Oh, there.
Hi. I wanted to ask if you are, for example, as a scientist approached by journalists or the press, the media, how do you ensure, because obviously they're looking for a story to tell, how do you ensure that when you're engaging with them that you're remaining true to the science and representing it accurately?
Yeah, that's a good question. So, I mean, a lot of journalists and media outlets will approach us if we're publishing a new publication. So we always ensure we're involved in the fact-checking process. A lot of the time you can't see the final article, But if you're working closely with those journalists, you can ensure that like all of the correct facts and science are in there. At the end of the day, you'll never see the final piece before it goes out. But a lot of the time, if there are any issues with articles, the journalists will let us go through and edit and even make changes to online articles. So, but one thing I would say is there are like specialist journalists, ones that are working in the marine conservation space or like environmental communication space. Those are the ones you want to work with because they have scientific knowledge and an understanding of the right way to communicate your work as opposed to kind of like mass media outlets. So it's about kind of picking wisely the journalists you want to work with. I can give you some recommendations of good ones after this talk if you like.
Thanks, Jasmine. Next up, we have Wade. He's going to be talking about decolonizing the terminology and language in marine education.
Good afternoon, everyone. Can you hear me? Goeiemorgen, Moluen, Ayubuvan. My name is Wade Nodi. I am the Community Program Coordinator at the Save Our Seas Foundation Shark Education Center. I need to say the full name at least once, which I will now be referring to as the Shark Center. And today I'll be speaking about our experience as an environmental education center and the use of language in marine education. I use the term decolonizing in my title because I think it's important for us to acknowledge the colonial influence on language, specifically in educational spaces and the field of conservation. I use the D in decolonizing because it's important for us to re-look and rethink approaches which have previously excluded local and indigenous communities, specifically in the field of conservation. At the Shark Center, our aim is to foster lasting positive connections to the ocean through education focused on sharks and local marine ecosystems. And we are based in South Africa, in the Western Cape area, at the bottom, specifically in Cape Town, Cook Bay. Our target audience are from coastal communities, mostly from under-resourced communities. As you can see, most of them stay very close to the coast, but that doesn't always mean that they have strong connections to the ocean. In South Africa, we have about 11 different languages, 12 including Braille, and we also have some languages that are not official, but they are still widely spoken within our communities. Most of the groups that visit the center either speak Afrikaans, isiXhosa, or Afrikaans as their mother tongue. And Afrikaans, which is seen as a dialect of Afrikaans, is widely spoken in the Western Cape area. And it's considered a creolized language, which developed amongst the interactions between the Dutch settlers, the Southeast Asian populations, which were brought to the country as slaves, and also the indigenous communities that existed there. So most of what we notice that most of the time when groups who visit the center, when they come and they don't speak English as their mother tongue, that we notice that they might not be as engaged as they could be, especially when like complex terminology and scientific terminology was used. And this was evidenced because there was little to no response and difficulties in communication, especially when, those big terms were used. In 2021, a social science study done by the University of Cape Town showed that when learners from under-resourced communities can feel alienated when visiting neighborhoods in previously white, when visiting institutions in previously white neighborhoods, which was classified as such under the apartheid system. And we believe that receiving knowledge in an environment unlike your own can be disempowering. And we believe that this might be one of the reasons why learners do not feel fully comfortable expressing themselves, which can also prevent them from engaging fully. So what we decided to do is we decided to use a language obviously a bit more relatable to our communities, and we decided to translate our presentations as well as our physical resources, which are still in the process of translation, and we also decided to change the language that we use when teaching. Through our translation process, we noticed that some words were a bit easier to translate than others. So for example, white shark, which translates in Afrikaans to wit haai. But we also noticed that in some communities, which still speak Afrikaans, that it might differ. So for some, it's known as groot doods wit haai, which translates to big death white shark. And when educating kids about the importance of sharks, I think it can draw a negative perspective of the species and can be a bit controversial, when educating kids about the importance of sharks. We also speak about the difference between sharks and fish, one of it being cartilage versus bones. And when we translated the word cartilage, if you put it into Google Translate, it becomes kraakbeen, which translates to crack bone. And when we crack as the act of cracking, like cracking your knuckles or your arms, and when we use it in our lessons, I was speaking in Afrikaans to one of our kids, and I was explaining to them that we have cartilage in our ear and the tip of our nose. And the kid asked me if our ears can crack, which kind of shows the dangers of direct translation and how some terms can be misinterpreted. In other languages, such as isiXhosa, if you translate it, it becomes, if you Google Translate, you'll get the other word, which is called inhlala. But when I spoke to a isiXhosa colleague, they told me that the kids might never actually use that word, but they have a different term that they use, which is huam. And that's actually the sound that the chicken makes if you eat the cartilage of the chicken. I don't know if you guys have ever tried that. Go like this, hum. Yeah. So that's the word if you put it into Google Translate. We also noticed that some terms were a bit difficult to translate or either didn't exist at all. In Afrikaans, the terms did exist, but even though they did, some of our communities or from our target audience would not use that frequently in their speech. So, even though maybe they might be exposed to similar terms in school, it's not something that they use in their daily vocabulary. And so for example, some terms can sound foreign to some of the communities that we engage with, which is why we just needed to be a bit more aware of that as well. Yeah, so after discussion with the team, we decided that the best way to deal with the complexities the complexities of translation with these terms was actually to meet the audience halfway and just to connect with the learners a bit more. Also to use multiple languages, give descriptions, but also to, just to be more aware and responsive to what was happening within the classroom. So we decided to listen to the learners more attentively, especially with what they already know about the ocean and the language that they use to describe these animals and processes. So for example, with sea urchin, a lot of times when we ask the kids what that is, sometimes they don't remember, or they'll give a totally different word. But if you directly translate it into Google, you'll get sea egel. I'm Afrikaans and I've never even heard about this word. I've even studied marine science, I've never heard about it. But this is what you put, if you put the word into Google, you get that. But in some communities, they also have their own way of describing the animal. So for Afrikaans communities, they call it a sticky ball. isiXhosa will say uhlaba, which then refers to the prickliness of the animal. So it's their own interpretation of what they see or how they engage with these animals. In some cases, for example, limpet, if you directly translate that, it becomes klip mussel, which translates to rock mussel. And some communities actually have never they don't know what to say or they don't know what it is. They just call it a snail, because that's how they've seen it. But there was one specific community, which is an under-resourced coastal community in Cape Town, from Ocean View. So they have lots of knowledge about the ocean. It's been coming down from generations. And when they came, they actually told us a name for this animal that we haven't heard before. It's called the limpo. They actually also they eat it, and we've actually never seen that before. So it was very interesting to also learn from the community itself. And when we say these things, or when we show them the picture, they know exactly what it is. But if we say the name beforehand, they wouldn't actually understand what you're talking about. But the knowledge does exist there. In Xhosa, it didn't exist. They would call it a snail. But some communities also know it as a perafuchi, which is actually just translation for a horseshoe or horsefoot. Also shows about how they interpret these animals and what they see. For some animals, such as sea anemone, if you translate it, it becomes sea anemones. At the end of each of the lessons, we do a reflection. So for kids to see what they've learned throughout. A lot of times they can't get to this name. And even Nemo, that lived inside of it, doesn't know how to pronounce it correctly. And so what they will do is they have their own interpretation. So in Xhosa, they say vulavala, which is open and close. And then Afrikaans will say sea tuti, which directly translates to sea vagina. Our goal was to connect with the learners, obviously, with what they already know about the ocean in the language that they use, and then to introduce the marine science terms to enable them to learn more. But in doing so, we needed to make sure that the terms doesn't sound too foreign to the communities. So for example, we also give descriptions for some of the animals. So when we speak, for some of the terms, when we speak about dermal denticles, we'll have to also explain in the language that's more relevant to the group. And then in some cases, they couldn't come to either the English or the Afrikaans word, so they'll either point to the side of the neck to indicate, like if they speak about gills, they point to the side of the neck, or they'll say lines. And in some cases, they couldn't come to either like the correct term, or they'll give it just a different term with a similar meaning. So one kid said the shark was invisible instead of saying camouflage. So we knew what he meant, but we'll then introduce the correct term and then explain to them a bit more about what it means. So also connecting them to them or meeting them halfway. Yeah, so some of our outcomes was that there was a considerable increase in the level of engagement. So the kids were a bit more comfortable, even if they made mistakes, they would still answer, which kind of showed a bit more of the confidence level. And also there was a lot of factors that played into this, but we believe that using a more familiar language could have assisted with this. The educators were also more aware of what approach to use if the educators can't speak the same language as the learners, whether it is to use the translated terms or either to use someone in the class to help with the translation for the learners. And also the learners were a bit more comfortable switching in between languages if they were able to do so. These are the recommendations. I think I'm almost done. My time is almost up, but these are the recommendations. Yep. But I would just like to say thank you to these people right here. That's my team and my supervisors.
Thanks, Wade. No, we don't have time for questions, so we're going to move straight on. Next up, we have Elisabeth. She's going to be speaking about connecting minds, hearts, and hands of ocean stakeholders for safeguarding marine biodiversity.
Thank you. My name is Elisabeth, and I was born and raised in landlocked Switzerland. And before moving to Bangladesh in 2002, I was trained and worked as a teacher and principal in a very small rural school in Switzerland. After moving to Bangladesh, I invested my skill set into building an educational outreach and marine conservation program, and including policy programs. And I hope that my experiences and insights gained over the last 20 years will motivate you to incorporate targeted educational outreach in your conservation plans, and also not to shy away from engaging with policymakers and enforcement officers. At the end of the 18th century, beginning of 19th century, Switzerland had a very large rural poverty area. It was widespread, and children were put to work at factories at a very early age. And their suffering and inability to help themselves guided a man called Pestalozzi to revolutionize the modern education principles. He believed that education was a right of all people, regardless of their social or economic background. And Pestalozzi's educational philosophy centered around connecting minds, hearts, and hands. And his approach emphasized the balanced development of intellectual, emotional, and physical capacities. And he combined this approach of academic learning, emotional well-being, and hands-on sensory experiences. He integrated physical and experimental learning into the curriculum, and such as working in the fields or learning crafts, and made learning more tangible and relevant. This holistic approach aims to foster critical thinking, empathy, and practical skills for a well-rounded education and a cultural engagement. Thanks to Pestalozzi, illiteracy was eradicated in Switzerland almost completely by the 1830s. To share understanding and knowledge is one of our core responsibilities as scientists. And today we all know and are aware that tools and approaches for affecting positive change must be sourced from multiple sciences. So think of all these sciences, and there are many more, as kind of languages, as we just heard. We don't need to speak all the languages perfectly. but we need to engage with people that speak different languages, that have a training in different sciences in order to enable the best possible solutions, including for conservation challenges. And working across these silos also applies to working in government sectors. Shark and ray management, as you all know, is between wildlife and fisheries, and in some factors, environment or home security. So it's multiple agencies, multiple departments, ministries, and institutions. And facilitating clarity and looking at the responsibilities of decision-making and enforcement, power, but also financial resources is really important to do across the government actors. It is challenging, but that should not stop us from trying. Different people or audiences require different approaches and tools, as we just heard so nicely. And to enable their engagement in conservation solutions, we need to be innovative and we need to speak different languages. Pestalozzi's principles show the importance of enabling experiences and meaningful connections to foster respect and appreciation. As we heard, it's really important that we not only clearly define our audiences and objectives and contents and resources, but that we actually know and respect our audiences as well. Think about it as languages. What is most appropriate for somebody who cannot read? What is most appropriate for someone who doesn't have time or doesn't care about the environment? These are all decisions that we need to think about when communicating in order to empower every decision maker to work towards conservation solutions. Engaging stakeholders requires different strategic approaches, and using protocols or guiding tools that enables you to replicate the efforts, enables you to compare different approaches, and very importantly, it allows to hold people accountable to what they're doing. So it's very important to see all the different tools that are already available and choose the one that works best for you as an individual or as an organization. One of my former colleagues, Russell, will be showing you how choosing these tools helped in Bangladesh to reach hundreds of thousands of people and bring about real change. Conservation management aims to safeguard a healthy ocean that continues to benefit us. And it is part of our responsibility to facilitate the collaborative development, implementation, and monitoring of good conservation solutions. So getting responsible sectors to coordinate and collaborate is the most effective way to use limited resources and maximize effectiveness. And when you institutionalize these intersectional collaborations, then you have the best chance of actually sustaining these. If it is in the mandate, if it is in the annual plan and the annual budget of different government organizations to communicate and collaborate, you have the best chance of making that a long-term engagement. And how to engage these people and how to learn about your partners. There are many toolkits out there and many good approaches. How to communicate the knowledge, the science that is required to make the best possible decisions. And remember, it's always a two-way communication. It's learning and learning from others. For that, I just ask that you use innovation. Learn to know your audience and to respect it, and then you will find the best avenues to talk to them, as Wade just said. And if you have the time and are so inclined, then I suggest you look at the pedagogical principles, because that is about how to learn to learn. Thank you very much.
Thanks so much, Elisabeth. We have time for one quick question, if anyone has. Okay, great. Thank you so much. Next up, is there a question? Oh, sorry.
Elisabeth, thank you so much for the talk. I'm just wondering if you could give one quick example of a way that you or two different agencies have institutionalized that communication.
The example that has worked best in Bangladesh is the CITES platform, because it is a legally binding agreement. There is international focus on compliance. And because sharks and rays are overlapping mandates between fisheries and forest department, we were able to negotiate collaborations across those. So for example, we have annual national CITES meetings where all the responsible agencies, and it's 12 of them, come together and share updates, steps, and decide on next priorities. That goes all the way down to the field. So when we do trainings, we train wildlife officers as well as fishery officers to recognize the species and know the regulations.
Okay, cool. Thanks so much, Elisabeth. Programming has resumed. Next up, we have Jamy, short and sweet, telling stories with science.
Okay, hi, I'm Jamy. I have a background in design, and I work with researchers to help them make the research feel more accessible and engaging to different audiences. So I'm just going to talk today a little bit about the fundamental things that I see as a story doing, as well as a bit of a process that I use to come up with a story structure. Okay, oh fuck, I meant to use it, sorry. Okay, so the thing about communication is that it's less about imparting information and more about understanding how that information will be received. To your audience, even though you need to relay the science, they're going to engage with your content for the same reason that they engage with anything else. Your communication is going to live in a world with Trump tweets and cat videos. And even though it doesn't need to be like them, it will need to feel interesting next to them. So this is why we use the term storytelling a lot. Okay, so effective science communication requires a little bit of a paradigm shift from I have all this research to share to what story does my audience need to hear about my research and how would they like to hear it? Okay, so I think storytelling is important, but I also think it's quite a confusing term. For instance, I use it to describe my work, and it generally looks something like this with not very many words. So what am I talking about? So instead of thinking about storytelling as any one form, I think about it as doing a few key things. I mean, it does a lot of things, but these are kind of the fundamental things. First, it acts as an entry point to a subject for a particular audience. Secondly, it delivers a message or messages. And lastly, it provides context for those messages. Okay, so entry point, I'm talking about accessibility. Your story needs to give your information a face or shape that feels comfortable and easy for your audience to interact with. Your audience should feel like it's meant for them. It should speak their language. It's really important in communication, especially science communication, to meet your audience where they feel comfortable in a format that feels familiar to them before leading them to new information. And secondly, an entry point is easy to follow. Basically, if someone is scanning through your communication, they should be able to very quickly pick up what it's about. A lot of this has to just do with ordering it very well, that it makes sense, and signposting, which is things like making clear sections, giving very clear titles, just basically like making the important information and the flow of information stand out. Something to consider is that most people don't like to read a lot. They like to look at the pictures to make sense of things. So if you pair signposting with relevant visuals, it can add a lot of engagement value. Okay. Okay, so the message. A message is not the facts themselves, but the conclusion that you'd like your audience to understand about those facts. And lastly, providing context. So you're giving your audience a bunch of information, but in order for it to stick, you need to give them somewhere that they should fit that into the way that they view the world. Give them a reason to care about it, and to remember it. So you can do that in a lot of different ways, by relating that information to something relevant to their lives, by using analogies when the science itself isn't that relatable, or just zooming out a little bit and giving them a few different reference points to anchor that information to. Our brains love patterns, so if you give a few different dots to connect, the information is going to feel more relevant and it's more likely to be remembered. Okay, so that's a basic checklist for what a story is, but how do we get there? So whatever form a communication is going to take, I always follow the same process to come up with a structure. And it sounds pretty simple and straightforward, but I think when you are sitting on the side of having done all this research, it can feel less obvious than it sounds. And that is, instead of working forwards from the point of view of I have all this research to share, it's to work backwards from the bigger picture, from what overall impact do I want this communication to have in the world? What impression do I need to leave my audience with in order to achieve that impact? What messages does my communication need to contain in order to make that impression? And only once you have a very clear idea of what those messages are, you can then go to the research and use supporting facts from the research to support that message. And this isn't about cherry-picking facts, but rather about being really clear on the overall picture that you want to paint for your audience with those facts. Okay, so again, it's that working backwards instead of working forwards is quite a subtle shift, but it's really useful in a few ways. First of all, when you're working with research, it's often very granular, and it's really easy to get bogged down in the specifics and the details. So if you have, what is my impact? What is like the overall goal of this? Just in the front of your mind, it just helps you really keep on track with reaching those goals, your communication goals. And secondly, oh, whoops, I wasn't meant to do that, but what I find most useful is focusing on the message before focusing on how to communicate the specific details. So for two particular reasons. First of all, I think accuracy is the biggest conundrum in science communication. And when you are stuck on thinking about, like, well, how do I communicate those specific data points, like, accurately, it can become very overwhelming fast, and it can also miss the point, because sometimes something can be really accurate on paper, but it isn't going to necessarily create an accurate understanding in your audience. So by focusing on the message first before thinking about communicating the specifics kind of avoids that, because messages can still be accurate while being really concise, and you can shape them in a way that your audience understands. And secondly, once you are very clear on those messages, going back to the research and being able to pick up the salient points that support those messages becomes a lot easier. It's a much more streamlined process. Okay, so in action, this was an infographic summary I did for the IUCN Global Status of Sharks, Rays, and Chimeras, the Save Our Seas, and in particular, Lauren DeFoss. And it was really important to follow this process for this particular piece of work, because the report was over 2,000 pages long. And so it was really important not to get lost in the details and the research, but to be really clear on the impact that we wanted and the messages that we needed to get there. So using the process, the impact that we wanted, mirroring the report, was action in terms of policy and implementation, and the impression that we needed to leave was sharks and rays are important, we should care about them, they are in trouble, and you can do something about this. So once we kind of had that impression in mind, it became clear that the message needed to lay out an argument that ends in action. And so at this point, I always just sort of map out the very, very general beats of the message. And then once I'm happy with that, I sort of expand it more with the general themes, the general relevant themes from the research. And once that flow is kind of making sense, I start to refine it and put it into some kind of wireframe, and start to work on like, how does this information flow? You can see at this point, there are no specifics. It's all about like flow of information and the overall message. And only once I have that structure, like really happy with how that structure works, then it's very easy to go back to the research and be able to populate it with the data that supports that message. So that's what the final sort of stuff look like. Okay, those were the two things I spoke about. Thank you very much for listening, and thank you to Save Our Seas for your support.
Okay, I'm going to not miss any questions. We have 1 minute for questions. Does anyone have a question for Jamy? Okay. No, thank you so much, Jamy. Okay, we're going to move on to speed talks next, just because we like to keep you on your toes. We have Iara next. She is going to be speaking about art, science, and sharks, reimagining public narratives through the Shark Stories Hub.
Hello, my name is Tiara, and my project is a storytelling platform. You know what? Never mind. That's the version that doesn't work, right? Let me try again. So everybody in this room knows sharks aren't the enemy, and yet that's a story many people believe. So the problem here is not how much we know, it's how we translate it and how we reach the people who actually don't know yet. So I tried a very simple experiment. I searched shark stories online, and almost everything I found was about shark attacks or poorly reported shark accidents. But the truth we all know here is very different, right? Sharks, they're complex, diverse, ecologically essential, very cool animals. And everything you do as a shark scientist is part of a story, just not the story people usually remember. So I'm probably not the one here with the best shark story. Maybe, does any of you have any moments with the shark that stuck with them? Yeah? Okay, well, that's amazing. And the problem is those stories barely exist where the public can find them, which is why I'm creating the Shark Stories Hub. It's a social media platform to challenge the limited narrative around sharks by gathering stories that celebrate their diversity, ecology, and individuality through creative storytelling. So it's a place for sharks, and not only shark scientists, to be the focus of the story. And I'm not gonna lie, at first I had many doubts, but at some point I stopped asking, will there be enough shark stories to tell? And I started asking, where are these stories hiding? And the answer is Sharks International. Because what better occasion to look for shark stories than the International Sharks Conference that takes place only once in every 4 years and is now happening just 100 kilometers away from me. And that is why I'm here. Because it's hard to balance accuracy and storytelling to avoid anthropomorphism or oversimplification while maintaining engagement. So instead of choosing between rigor and impact, why not build both together? Why not have scientists, communicators, artists collaborate to purposefully and responsibly shift this narrative about sharks? So the stories on the hub are built as short, visual, mixed media carousels featuring real-life events or stories, published science, or introductions to lesser-known shark species. There's also the chance for storytellers to contribute scientifically accurate fiction stories that will be clearly labeled with that tag over there. And this platform also provides a chance to cover shark news in a responsible, science-backed way. So the Shark Stories Hub is a space to create a community between scientists, artists, communicators, so that we can all work together to shape the narrative around sharks. So if you've ever had a special encounter with a shark, if you've published something that you think deserves to be seen beyond academia, if there's any species or behaviors that people should know about, please reach out. And if you know people who might have cool stories, please help us connect, because those are exactly the stories that this platform is meant for. It's a space for all of us to put together all the stories we already have so they can finally compete with the dominant narrative. Thank you very much.
Yara, does anyone have a question for Yara? Quickly, there we go.
Well, first of all, this is super cool. My question is it only going to be written stories? Are you also doing video or audio stories?
So that's a good question, thank you. The truth is that right now, this is a project in development. And the reason that I'm presenting it here is because I'm hoping that the shark community will help me develop it. It's also a big task to take on, you know, to want to centralize all these stories. So for now, I was thinking about it as carousels, just because I like illustration and it was kind of more the part that I could do myself. But ideally, I would love to have like other creators in the community collaborate in the hub. So it would be amazing to also do video format in the future, or even combine, we can do like video and photo carousels together. Thank you.
Okay, thank you so much, Yara. Next up we have Paul. Paul is going to be talking to us about the Speaking Up for the Oceans Toolkit, how to communicate problems facing sharks and marine life, and to make change seem possible.
Hey, comms people, how are you doing? Sorry, I've missed a bit of this session because I've been moderating another session, so I hope you had a great time. Is this the clicker? Okay, so I have 4 minutes to talk to you about 5 traps that could be standing in the way of the one thing that we all need to be doing for sharks and rays. Communicating with impact. It's not just what we say about sharks and rays, but it's how we say it. That's what's going to enable us to connect with more people and to gather more people towards our cause. So to that end, we've published the Speaking Up for the Ocean Communication Toolkit. It's for anybody who wants to speak up for sharks or just want to speak about sharks and wants to be guaranteed to have impact in what they're saying. In the development of this toolkit, we looked at literally hundreds of websites, social media posts, and working with framing matters, we looked using social science principles, we assessed the messaging for impact and for language. And the result of this work is 5 communication traps that we all need to be aware of and conscious of. So what are these traps? Here we go. Number 1, the sea otter trap. Scientists beware. Not everyone loves sea otters. Some people think sea otters are cute. Other people think they're a pest. And likewise, not everybody understands the meaning of our words and our language in the way that we do. So burst your bubble, drop the jargon and the acronyms, speak in clear, everyday language. And remember, it's not the right word if the people you're speaking to don't know what you're talking about. The second trap, the flatfish. Likewise, are we using words and language that are losing their impact because they're disappearing into the background? When we use terms like overfishing, bycatch, habitat degradation, are these words hitting home, and are they making people want to do something about it? So when we're talking about issues, make sure, again, we speak in a clear, everyday language about what is happening and why. Third trap, don't be lazy like a sea cucumber. Don't forget to tell people the cause of the problem, and don't forget to not use hedging language. So when we're talking about what can change and what can happen, don't use hedging language or caveats that just make you sound uncertain that you're going to be able to achieve change. We are not striving to try to make a change to things in the future. We can change this. Be positive, and people will follow you. But, and I think we all know this, don't overdo threat. Too much threat makes people want to hide from the problem and just ignore the issue. This isn't about us shying away from the problem. It's about presenting the problem in a way that it appears to be solvable. Talk about past success. Talk about past successes. Talk about the progress that we're making, and never present a problem without a solution. And finally, this is really last but not least, and this is where I'm expecting tomatoes to be thrown at me. The octopus trap. Don't mimic language. What I'm saying here is let's do away with the myth-busting. When we talk about something and we try to play it down, often we're just reinforcing what we're saying. It's time to drop the coconuts, the vending machines, and the toasters. Let's just come out positively with sharks. Let's all put our stories on the Shark Hub, and let's stop being so defensive and find ways, develop techniques for twisting the conversation, turning the conversation to what you want to talk about, not to what they want to talk about. So great. How do we deal with all these traps? Well, the good news is it's all in here. There's our pitfall, all the pitfalls and how you can overcome them, some suggestions for you, as well as a whole bunch of tips on planning, delivering, and evaluating good conservation communication projects. So to download it, Shark Trust Toolkit, just put that in your search engine. Let's talk sharks.
Okay, great. We do have, it's Tina. I don't know if anyone has a question for Paul. Let's keep him from the T. Does anyone have any questions? No, excellent. Off you go. So up next, we have Raquel, and she is going to be chatting to us about evaluating the paper of science communication in raising awareness about sharks. There we go.
Okay, so hello everyone, my name's Raquel, and this is my master thesis in the History of Science and Science Communication Master Program. And I'm going to talk about a project that I did in science communication, because as everyone in here knows, the way that we talk about sharks shapes the public perception that people have about them, and we only need 2 musical notes in order to prove this. Because with just 2 notes, we can change the view of a sea, a normal sea, into one of our worst nightmares, just thanks to the John Williams music of the movie that we all know, that it's Jaws. So, the power of communication can shape reality, and it's a great tool in order to villainify these animals, but also in order to change the public perception and make a paradigm shift that these animals really are in need of. So, however, for communications to be effective, it must be rigorous, and when we are constructing a science communication program, we must be well aware of these 3 limitations, such as reported by Ziegler and the team in 2021, and these limitations are that often science communication initiatives lack of precise objectives and control groups, and that makes it really hard to evaluate the effectiveness more often than saying raising awareness about sharks, and we really need to change the way that we select the objectives of our science communication initiatives. As well as this, another issue with science communication projects are the methodological issues in evaluation, because a lot of projects do not use statistical tools in order to measure the effectiveness, and that makes it like hard to believe for other scientists, or it lacks of rigorous approval. And also, we find limited transparency and evaluations because the evaluation tools are not shared with everyone, and it makes science communication look like it's only success stories. And we really need to change this perception, because science is made of failures also, and we need to know what's wrong and we can do in order to change it. If we only communicate success, we will not learn enough in order to make science communication rigorous and scientifically profound. So, in order to make this project, the Proyecto AtracTibu, we address these limitations by doing some kind of things that I point out in order so you can make it too. And we define objectives, clear objectives. We group them into categories. Yes, it was raising awareness, but beyond raising awareness, we need the people to know what is a shark, how do you distinguish a ray from a shark, and we have clear objectives, as well as a control group in order to make a statistical analysis and to see if really the experimental group was learning something from the project. In order to address the methodological issues in evaluation, we use statistical analysis using ANOVA and Welch's ANOVA in order to measure the effectiveness, and then statistical analysis such as the T3 Donnet test and the Bonferroni test. And we moved away from auto-reported data in order to not make it like biased by the people that were conducting this project. And in order to address limited transparency, we decided to give free access to all information, and you can find the evaluation, the scoring criteria, everything that was made in the project is available and free for everyone to check out and to compare if what we are saying is truth or not. So, for this project, the Proyecto AtracTibu works with one of the most dangerous animals in the world that has terrorized adults for years, and those are teenagers. In the project, it was a service learning project, and the students carried out some different activities in order to learn more about sharks. We didn't do like classical learning with classes. We moved away from traditional learning methodologies, and we did something more interactive. For example, they came to the facilities, to the oceanographic, to do a guided visit to the sharks facilities all around. Those are the girls and the boys from the project, and they knew sharks a little bit closely. They also did interviews to people that work with sharks. This is Jaime Penades, that is sitting right here. And the student had to be capable of doing an interview to a scientist, to a conservation specialist, and these interviews are recorded, and they work with them and uploaded on the YouTube of the high school. They also, like, the heart of this project is the in-class breeding of Styliorhinus canicula small spotted cat shark eggs in the classroom, and the students took care of these eggs until the sharks born, and then we retrieve them and they grow up at the oceanographic facilities. But the first stages of the hatching progress, they did it at the high school, and we talk about this later, but this was like a really important tool to make an emotional connection between teenagers that were really scared about sharks and making them care deeply for these animals. We also did some more activities, such as a dissection of bony and cartilaginous species in order for them to see practically engaging with these animals the differences between them, not just learning from the hearing. We also, in order to make transversal this project, we collaborated with a local artist that made this commemorative mural in the high school in order to involve the children from the art classroom and make it all more accessible. And then this was really cool because the students that were learning about sharks, then they became the shark teachers, and they made projects with children and explaining the project to other people. And we did some events also in the municipality, and these are the results of the statistical analysis. We found statistically significant differences between the evaluations carried out in the second phase after the project and 1 year before the project completion. So we can say that statistically there are differences in students' knowledge and perception of sharks that lasted long term, and we will talk about the implications later. We also, in order to make everything, I don't think the video is working, I don't know how to play it, but in order to make this information available, we have a website with all the resources and all the information for the project, such as this one's also, that it's like an online guide of sharks that is accessible to everyone, and a shark booklet with activities that the students carry out in the high school. And the key takeaways of this project is that we need to make learners advocates for sharks, and that we had a significant long-term learning and awareness. It lasted 1 year before after the project finished. And we have also statistically proven effectiveness and high satisfaction and replicability. In the future, this project has already been replicated by the Fundación Azul Marino in Castellón, in Spain. And this is some of the bibliography, but I want to leave you with this slide because you can find all the resources and all the information about the project in this QR code. Also, I didn't bring the stickers, but tomorrow you can find me. I'm very recognizable because of the streak of hair and the eyeliner, so come find me and I will give you a sticker of the project. And in my link tree of the Instagram, you can find my full master thesis if you want to read it or check out the data. And that's it. Thank you for your attention.
Okay. Thank you, Raquel. Do we have any questions for her before we move on to the next talk? Okay, perfect. Thanks, Raquel. Next up, we have Anna, and she is going to be talking about Putting Patrol, building a nationwide network of shark advocates.
Hi. I have a support team. Good afternoon. I'm Anna. I'm the Executive Director and Chief Mermaid of Save Philippine Seas, and I'm going to be talking about a project called Putting Patrol. Putting is shark in Filipino. So this is a project in partnership with Marine Wildlife Watch of the Philippines, and its founder is here with me, AA. So what are the objectives of Putting Patrol? First is to increase the knowledge of our participants. These are community leaders, they're enforcers, they're dive operators, and increase their skills in identifying shark species. We also want to build a nationwide database of shark sightings through citizen science, and develop a nationwide network of a sharkada in important shark and ray areas, or ISRAs. So in Filipino, the word sharkada means, barkada means your group of friends. So these are people that you grew up with, probably you went to school with. So we wanted, I'm obviously big on wordplay, so we call them our sharkada. So in the first year, we did 3 workshops, and they were all in ISRA sites in Batangas and Palawan, and we had 108 participants. Majority were male, which isn't surprising because the enforcement field is male-dominated. But we also had majority of local government participants, which is good because in the Philippines, the mandate of managing municipal waters falls under the local government. So this is a one-day program. It's a very packed program. It starts with why sharks, why we need to care about sharks, what their reproductive features are, how they are threatened by different issues in the Philippines. And then what about sharks? This is the legal frameworks, and we present it with a timeline of current events and pop culture moments in the Philippines so that people remember. And then we have Are You a Shark?, which is our shark identification module. And then we just added this one. It's what do you do when you get a protected or not protected shark? Do you turn it over? Do you report it? Do you let it go? And then finally, we have a lecture on ISRAs, which really cultivates people's pride in their location. And then we have a commitment setting activity in the end. Now, every single activity has something fun. So growing up, there was this famous show in the Philippines called Battle of the Brains. And so we made it Battle of the Brain Corals. And this is a trivia game. And everyone gets really competitive and has fun. And then we have Pin the Fin, which is inspired by pin the tail on the donkey, but we made it pin the fin on the shark. So this gets everyone, again, super competitive. We also have, of course, cute merch, because who doesn't love cute merch? We have shirts, postcards, stickers. We also give a set of protected species posters to all the organizations that attend, and they can display this. And we have a handbook in English and the local language, which is very important for people who aren't really fluent in English, to make sure that they have the lectures in the local language. So the way that it's designed, it's very interactive. There's a lot of laughter, a lot of music, and our participants always say that it's fun, it's informative, and what matters to me is that it's welcoming to people who don't have a background on shark biology or marine science. It's also entertaining. And we do a pre-test and a post-test, so the average score at the pre-test is 3.7, and then in the end, it becomes much higher, 9.4. And 100% of the 108 participants showed an increase in knowledge. There's also successful shifts in perception. So we ask them, what are the 3 words you associate with sharks at the beginning? And a lot of the words here revolve around fear, danger, they eat people, they should be eaten, they attack, they bite. And then at the end, we ask them again, and they're more positive or at least more neutral. So they describe the features of the shark like powerful, endangered, important. The word in the middle, pangalagaan, is like to take care or something that should be taken care of. We also launched a Facebook group, the Shark Spotted Philippines Facebook group, to collect shark sightings. Majority of the sightings are actually by AA, so that's he's the one reposting when he sees. And after the first year, we asked 20 organizations to make commitments. And a year later, we ran after them, we pestered them to ask, have you actually done anything based on the commitments? And we're proud to say that most of it was actually reached, although of course in varying different levels of success. So Shark Spotted PH, we already have 260 entries of dead and alive sharks. We also have different posters. They've actually posted the posters, so you can see here. There were viewing guidelines that were developed for the blacktip sharks in the ISRA. But there are, again, always challenges. Even if there were a lot of successes, there's still a lot of challenges. A lot of the organizations did conduct post-event lectures to their communities, but they're one-off. So we don't know if the information can be sustained. And it's not their expertise, so they can only re-echo what kind of presentations we give them. It's not necessarily up to them to make it deeper. And a lot of when we went back to them and asked them what their challenges are, they said, we don't have dedicated staff. The leadership changes in the Philippines very quickly, every 3 years. So you could be working with one passionate person and then they get kicked out or someone else gets elected. But what's next is really exciting. We are able to support the participants on their commitments. We're developing a shark conservation legislation toolkit to help local governments develop their own legislation for sharks. And we're developing more fun information materials. We've already done 2 more Pating Patrol workshops, and we have 1 more to go in June. And we just secured a 3-year project to expand and establish marine protected areas in ISRA sites. So if you have any questions, I'm excited to start marine conversations with you. Thank you.
Okay, that's well done. We have quite a bit of time for questions, so if anyone has any questions for Anna, please raise your hand.
They're speechless.
Jaws to the floor. Oh, that makes so much sense at a shark conference. Okay, yay, we've got one question over.
Hi, it was a great talk, thank you. I arrived a little bit after, so I didn't see the beginning. Like the people that you talk, like how are the ages? Like you get most children, most adults? Do you have like a difference between the perception of them when you do this test between children and adults and stuff like that?
Yeah, that's a great question. So this particular workshop is for, let's say, mid-career. They're probably, they're employees of local governments. We have another one on sharks. We have a baby shark storytelling session, and then we also have this program called Anak ng Pating, which is, in the Philippines, there's this expression, Anak ng Pating is like the child of a shark, and it's like negative, but then we want to turn it into something positive. And yes, we do use the baby shark song, unfortunately, but that's part of the fun.
Okay, cool. Thanks, Anna. We have a question over here from Wade.
Hi, thank you. Amazing talk. I just wanted to know your process of identifying the audience, or is it something that's how long it's been coming along? How long it's been coming on? Do they reach out to you guys? Do you guys identify them, and how does that work?
We reach out to them. So we partner in the different Easter sites. We have a local partner. So it can be a nonprofit organization or the local government there, and they help us identify who should be invited. And we have to make sure that there's a local organization because people don't respond to emails. So we need someone to physically go to the offices, give a hard copy, like a printed copy of the letters. But they're mostly local government employees. So they're working in fisheries or enforcement or local government planning, that kind of audience.
Okay, I've got another question.
Hi, can you tell more about, in the end of the workshop, you mentioned you make a pact or like a, what do you call
Commitment.
it? Yeah,
Yes.
commitment with the participants, and we saw like they actually carry through. Can you tell us a bit about this commitment? Was that, what is it like?
Yeah, we asked them to write what they can do for sharks and rays in the next 3 months and in the next 6 months. And there's a questionnaire like, what is your organization good at? And what do you enjoy doing? And then try to marry that. And then we had to include in our design, in our project design, to run after them afterwards to make sure that they're... But we also asked them, what kind of support do you need? If you didn't meet your commitments, how can we best support you? So that, we're also helping them reach their goals.
Okay, that's us for questions for now. The next talk is Mohammed here. Okay, cool. Okay, there we go. He's going to be speaking with us about engaging fishers and coastal communities through educational outreach exhibitions to promote shark and ray conservation in Bangladesh.
Hello. Good afternoon, everyone. So, I'm Russell from Wildlife Conservation Society, Bangladesh. So, today I would like to present our educational outreach effort to protect sharks and rays. Sharks and rays in Bangladesh. I have been working with WCS since 5 years, and I engaged in these community exhibitions. I spent months of times with fishing communities, and I experienced that inspiring and empowering fishers to protect threatened sharks is very important. So, Bangladesh presents a complex conservation settings. So, 40 million people in Bangladesh live in the coastal region, and among them, their livelihoods mainly depend on the marine fisheries. And so, what we did in this, there have some lots of fishing pressure, and as well as the sharks and rays, the priority habitat of sharks and rays are in this region. So the priority habitat of sharks and rays, and as well as the primary fishing ground, both are merged in the region. So this is a challenge that finding the solution to address these two challenges. So this is what we try to address in our outreach events. So to respond to this, our diverse aquatic ecosystem of Bangladesh, including its vast river network and mangrove forests and the coastal shallow waters and the deep submarine canyon, they hold more than 100 species of sharks and rays in Bangladesh, but more than 2/3 of them are threatened, that are worse than the global scenario. So this is the challenge we are trying to solve, that there has some lots of pressure like overfishing, high bycatch rate, and the local and international market demand. So we try to solve these critical challenges for the slow-growing animals. So what we did, to respond to these challenges, we initiated educational outreach exhibitions guided by a communication strategy and a theory of change to bring positive shifts to the fishers in their knowledge, attitude, and practices. So we believe if fishers comply with the regulations and safely release protected species, if the traders they understand and follow the regulations, and if the consumers they can recognize the species, what to consume or not, and the local government officers, they can know about the laws, national and international, and to comply and enforce in the ground. And the policymakers to will to the policies to change for this, to conserve these threatened species. So if that all will make change, then will be found a healthy ocean for these threatened species. So then what we did, we translate our complex science into simple, meaningful messages, like that they can understand, and this is their daily uses, they can use the things. So we are linking unfamiliar species, like if we told them species, they will not recognize. So we try to simplify this so they can attach with the species, and they can feel it relevant. Like we use saw for sawfish, and like a pair of pliers for manta ray, sickle for thresher sharks, kind of things. So this makes identification easier. So they understand which species is protected, nationally or internationally. So then we have guided tour, like this is the consistent way to first to last, that we can comply the message so they can inspire and motivate to become an ocean guardian. Like we try to explain that why sharks and rays are important, then what the threats they face, and the rules and regulations of government and international, and how to safely release these species in the ocean. So we did educational outreach exhibition. This is one of the initiative to protect mainly sharks and rays. These are mobile, cost-effective outreach program. We bring it to the landing site and coastal harbors. There are lots of fishers and traders meet every day. So, and we make it engaging and interesting, fun way, so they can understand, and as well as they will make it as a fun and memorize these things. Like we first announce the event before our exhibition. Then we guided the exhibition through our university students. They are try to make it simple to the community people, and then we try to engage it like lots of games you see in the pictures here. So lots of games and documentary film screening and lots of things, so they can engage with the things and learn from here. So then this is our another initiative, like Healthy Ocean, Healthy People. So this is the broader context. So we, Ocean Guardian, we did for only sharks and rays. This is for the marine megafauna as well as sharks and rays. So we did this like our, in Bangladesh, major coastal landing site. So we engage till date like 60,000 fishers, traders, law enforcement agencies, and community peoples to inspire them about this marine wildlife. So for this, we use highly illustrated and visually compelling outreach tools. Like you see here that lots of outreach tools here. So we distributed like 100,000 copies of these outreach materials in this time period, so they can understand which species is protected and which are not, as well as we install them in the their high frequency areas like tea stalls and traders office, like fishing landing site and fishers hub in that area. So what is our outcome? So that's why we did a pre and post evaluation, and we see this, that overall 35% increase in their knowledge and 68% knowledge they increase in the protecting identification of protecting species. So we have another network, Citizen Science Fisherman Safety Network. There have some lots of activities. They plus 60 plus incident that they like safely release the our sharks and rays. So I have seen you the last video. So this is our exhibition, and they inspire to safely release these species in the water.
Marcel, we're going to have to... Okay, we're done. Okay, yeah, we're done. So, okay. Sorry, I feel bad, but we've got to stick to time here. If anyone has any questions for Marcel afterwards, you can speak to him after the session. Cheryl is up next. She is going to speak to us about growing ocean literacy, shark and ray education program in the Seychelles.
Thank you for the support. Good afternoon, everybody. My name is Cheryl Mamo. I work for Save Our Seas Foundation with Island School Seychelles as part of the education and communications team. I am here today to present a project that Island School Seychelles oversees, which is called the Daros Experience. I will be referring it as DEX throughout this presentation. We define ocean literacy as the understanding of how the ocean works, how it influences our lives, and how our actions impact it. In short, the DEX experience doesn't just teach us about the ocean, it helps students understand it, value it, and feel responsible for protecting it, which is the core of ocean literacy. So what is the DEX program? So in a nutshell, the DEX program is a marine education competition that is launched annually for young Seychellois between the age of 10 to 14 years old. After an intense judging process, the participants, 16 final participants are then chosen to visit Daros Island and St. Joseph Atoll, which is where the Daros Research Center is based. Essentially, the participants have to submit entries reflecting given themes of each year, and the entry categories varies from art, literature, and as well as videos. So how does the DEX program actually links with raising awareness for sharks and rays? So as you here all may know, is at the heart of the foundation is the conservation of elasmobranchs. So the DEX program actually provides these students, especially at a young age, with a rare opportunity to visit an ecological hotspot, which is the Daros Island, where these species are found. They also get the opportunity to interact with scientists and researchers that interact directly with these species on a daily basis. They also get hands-on experience by interacting directly with these species. This helps them understand the species, and we hope also gives them a better appreciation of the species. This year, the DEX program is celebrating its 5th year anniversary. And over the 5 years, we've been able to track a few trends and patterns. One of them being, we've seen that we have a 58% of engagement from schools per year, with entries or applications ranging from 60 to 70, with only a high peak in 2025, where we received a total entries of 91. So far, we have received a number of 380 applications in total, with 12 reapplicants that came back for a second trial, and only 1 applicant came back for a third time. Out of the 12 applicants that actually came back for the second time, 7 of them were successful in being part of the DEX program. Learning, so the program running for 5 years now. So one of the key points has been to learn and adapt from the program as we go. And looking ahead, one of our key priorities for the upcoming 5 years is to be able to understand and assess the long-term impact and effectiveness of this program. We started piloting survey questions to understand this. So one of the questions that was asked was if the program actually improved their understanding of the marine environment. Out of 18 responses so far, 94.4% said yes significantly, 1% said yes somewhat, and no individual indicated no change. We also wanted to see if the program was changing their perception on sharks, because in Seychelles, sharks are often feared or also seen as food. 94% said yes positively, and 1 individual indicating no change. We also wanted to know which of the topics they now understand better around sharks and rays, and the majority responded with all the above, being the ecological roles of sharks and rays, threats facing sharks and rays, species identification, research, and monitoring methods. These questions we believe are highly important, for these will serve as guidelines as well as give us insight on how to better improve the program as well as assess its effectiveness in the long term. We do not want the DEX program to be a one-off engagement with these participants, which is why back in 2023 we have introduced the alumni program. Outside of keeping these students engaged, it also helps them gain a broader experience outside of Daros Island and Save Our Seas Foundation. Different outdoor events are organized, being ongoing swimming classes, snorkeling, hikes. The events varies, but it allows them to we also collaborate with other local NGOs. I am proud to present this because this is one of the projects that was held back in 2025, and it was done to celebrate Shark Awareness Day. It was one of the first of its kind done in the Seychelles, and it allowed students to do research around sharks and rays and then design costumes and parade it around the main island, in the main town of the main island. We were highly encouraged to see a high number of participants of DEX alumni. Just a few pictures to show you how cool it was. To conclude, I would like to leave you with 3 key takeaways. Initiatives such as these program plays a crucial role for conservation. Teaching the younger generation to value and appreciate these species at a younger age what will actually help conservation of the species in the long term. Data and science help us tell many beautiful stories, but if they go unheard, then their value is lost. The aim of this program is not to turn every participant into a marine biologist or a conservationist. We also want individuals, whatever career path that they choose, be it engineers, doctors, or even bus drivers, to have an environmentally conscious mind. And we want them to become young ambassadors and advocates for the marine environment as well as the environment as a whole in the present and in the future. Lastly, for me personally, this program has showed me that teamwork forms the foundation of conservation. It is the driving force that carries our efforts forward. This program would not be as successful as it is if we did not have the right support. And with that said, I want to extend my gratitude and heartfelt thanks to my team, to our team at Save Our Seas Foundation, the Daros Research Center, as well as Island School Seychelles. It is your passion, dedication, and drive that gives this program life. Without your hard work, again, it would not be where it is today. Thank you.
Thank you so much, Cheryl. We have time for one quick question. Does anyone have a question for Cheryl? Excellent. If you do think of a question, you can chat to her after the session. We're moving on to our speed talks now. Next up, we have Gillian, and she is going to be speaking about changing the narrative using photography, storytelling, and language to transform public perceptions of sharks.
Hello and welcome to my presentation where I walk you through how photography and visual storytelling can change people's perception of sharks. My name is Gillian Shark. I'm a shark photographer and educator with a background in business and education. I also trained with Swiss shark behavior researcher Dr. Eric Ritter in 2017 and 18. But I wasn't always a fan of sharks. Growing up in Switzerland, I was terrified of them. Until on my very first scuba dive, I saw my first shark, and I realized that they were so different to what the media portrays them to be. That's when my fear turned into a fascination. 10 years ago, I started with underwater photography, specializing in shark photography, because I wanted to show people the way I see sharks, as beautiful, majestic, and elegant animals. When I returned from my travels, I showed my photos to family and friends and posted them online, and the feedback was only positive. People told me that thanks to my photos, they were less scared of sharks, and that's when I realized how important and powerful photos are. Sharks are polarizing animals, and for most people, the understanding of sharks comes from headlines, media, and films, and not from real life encounters. Once a perception is there, it's difficult to change it, but I'm proof that it's possible. Not only images, but also the language we use shape our perception of sharks. If you use the word shark attack, it suggests intention and malice. But if we describe the same event as a shark accident, it creates a different understanding. The same goes for words like aggressive or shark-infested waters. And it's not about talking about sharks as docile animals. they're still apex predators, but it creates a different understanding and puts everything into perspective. There's a pool of evidence that shows that we only protect what we care about. And that's where visual storytelling comes in. Personally, I use photography to build that first step and to engage in a conversation. A shift from fear to understanding is what makes conservation possible. And if fear is something we've learned, it's also something that we can reshape. As a little takeaway from this presentation, I want you to pay attention to how people talk about sharks outside of this shark conservation science bubble. And maybe next time you hear someone say shark attack or shark-infested waters, you can tell them what you learned today. Thank you so much.
Okay, great. Thanks so much, Gillian. Do we have a question? We've got time for questions.
That was a great talk. I was wondering, do you feel like, what role do you feel like you as a person play in your photography when it comes to changing other people's perspectives? Like, do you feel like the picture in and of itself alone could change people's perspectives, or do you feel like the fact that you are the one behind the camera and they're talking to you clearly in person with all your limbs, if that actually has an effect?
Thanks for the question. Great question. And I think it's, first of all, the photos, because I tried to show the animals, the sharks, in a very elegant kind of way and not like the way we usually see sharks with their mouths open. But I also feel like it's my passion that kind of, makes a difference, because every time I hold a talk, especially in front of kids, their eyes light up when I show them photos I took. They go like, wow, did you take that photo? And I think that, enthusiasm definitely makes a difference. Yeah. Thanks.
Okay, great. Thanks so much, Gillian. Next up, we have Nathaniel. Oh, excellent. So Nathaniel will be chatting with us about, well, his topic is From Nursery to Narrative, Communicating the Science of Shark Rewilding.
Good afternoon, everyone. Just quick show of hands. Who knows about ReShark? Yeah, that's awesome. If not, for those who don't, I hope to take the next few minutes to share about how shark and ray rewilding is not just a scientific or conservation challenge and opportunity. It's also a science communications one. So at ReShark, we're an international coalition with a global mission to rewild our ocean's threatened sharks and rays, with our first and flagship initiative being the Stegostroma tigrinum augmentation and recovery project. And a whole lot goes into it, as you can see. From husbandry to veterinary care, to research, to governmental relations, and to repopulation and post-release monitoring, all of this work is very complex. And while the eventual releases of these amazing animals sure may seem like the sexiest part, a lot more work also happens behind the scenes. That can create barriers, making it difficult for the public to understand or support. Thankfully, shark rewilding is as much people-centric as it is scientific. We find that people are engaging with moments rather than processes. A hatch, a release, a resighting, a scientific breakthrough, or a new aquarium partner coming on board. These moments make complex systems visible and emotionally accessible. Second, visibility itself becomes a conservation tool. Our in-situ facilities, for example, are not just for conservation. They are visibility infrastructure, making recovery processes observable and communicable. And third, people trust people way more than institutions. In a distributed and diverse coalition like ours, storytelling comes from our aquarists, our field researchers, and our local partners. Leaning into this decentralized mode of operation helps us build authenticity and allows communication to scale beyond a single institutional voice. So instead of treating science comms as an add-on, we try our best to design it into our conservation process itself. We're shifting away from one-off earned media towards continuous owned channels, enabling longitudinal storytelling rather than just isolated moments of outreach. And in today's attention economy, dominated by vertical shots, we use these formats to our advantage. We're also constantly nurturing a culture of storytelling across our network, allowing as many voices to contribute inclusively. And I'm sure my colleagues have gotten used to my constant badgering to film themselves and to film everything. But doing so allows us to hear about all aspects of resharking, from the very people hands-on and knee-deep in the work each day. Shark rewilding inherently involves complex language as well, so we work to simplify and standardize how we talk about it, using accessible lexicon, digestible metrics, and a clear framing of the science. And over time, we've found that science comms functions very much as a feedback loop. Science and species knowledge enable conservation such as rewilding. Rewilding generates ample storytelling opportunities. Stories drives concerted engagement, and this feeds back into new knowledge, helping us refine project protocols, expand across ranges, and explore new candidate species. Our work hence reflects a shift away from one-way communication. We are no longer just audiences of ReShark's work, we are active participants in it. And the result hasn't just been increased global awareness. From school children to local fishers to international visitors and to governments, we are witnessing active buy-in, participation, and community stewardship. People are tangibly interacting with resharking. And it's been incredibly encouraging to see our work evolve from initial ideation to a scalable model across more regions and more species in less than a decade. This highlights the role science communications has and will continue to play in expanding our collective conservation impact. Thank you.
Great, thanks, Nathaniel. We've got time for one question. There's a hand at the back there.
Thank you. That was a wonderful presentation. Thank you. I was just wondering and curious if you guys ever put tags on some of the sharks that you rewild after putting them back into the ocean.
Yeah, we do. A lot of it. So follow ReShark. We are everywhere. We track the journeys of the tagging, and we track the journeys of the results that the tags inform us about the species. Yeah, follow us everywhere. You'll see what we have been doing.
Beautiful. Oh, thank you.
Great. Thanks so much, Nathaniel. We only have time for one question. We're on to our last speed talk of the day. Is Livy here? Yes, great. Okay, so Livy is going to be chatting to us about empowering conservation action, development and application of a multilingual visual guide for protected elasmobranchs in India.
Yeah, very good evening to all. Yeah, I'll be presenting on the topic how the complex laws governing in our nations can be converted into simpler forms with the help of some multilingual guides. As a country like India, having multiple languages, it is very important to have such kind of regional specific tools like science communication tools needed for supporting the conservation in India. So let me start. So the core challenge in any country, basically in India, we have a coastline of about greater than 11,000 kilometers. And also we have 9 maritime states, and each with a specific language. It's multi-language. And the fishing community, that's the population, is having a diverse culture. So we need to look upon to all these aspects while giving the conservation measures or while converting our laws. As you all know, most of the laws are very complex. So if any complex law, it has to be, the first and the basic thing is that has to be understandable by the end users. Here the end users will be officials who are doing the enforcement, and the fishers who are, so these are the two people who are coming into this. India in 2022, our Wildlife Protection Act is amended. Earlier, we have only 10 species of protected sharks and rays, but after the amendment, we have about 26 species which come under the Schedule 1 and Schedule 2. So there is a basic difference between these schedules. One is a bailable offense if you do some trade or capture of these fishes, basically this sharks and rays. And in Schedule 2, it will be like a non-bailable offense, but almost the other penalties, and it will be mostly similar. And also, we have a Schedule 4 in which the CITES listed species, protected species, are also included in these schedules. So it has national legislations we have included with the CITES frameworks. So let me introduce the guide. This is an illustrated guide of protected elasmobranchs in Indian waters. So as a result of these amendments, what happened means there is a confusion exists between the officials like enforcement agencies as well as the fishers. What they mean is most of them not able to identify the species. Earlier we have only 10 species, but now more than 26. So a basic thing is they need to identify which are all the species coming under protection and the laws behind them. So we have introduced this guide. We have almost 34 illustrations, and most of them are real photographs from the field itself. They are realistic photographs, and some of the rare encounters or say rare in the fishery, we have illustrations, that is detailed drawings. And in each page, we'll have the authoritative list like WPA status, that is the Wildlife Protection Amendment Act status, CITES listing status, IUCN, and the NDF listing, that is non-detriment finding status. And we have converted this into all the 10 regional languages. So here's what I have already told. And finally, we have, for the last couple of years, we have gone through many awareness workshops and talks. And finally, we got an output like they are more, they can easily identify the species, the officials, and the fishers, they are also coming up. So it helped them in decision making, like whether this, actually the laws are very complex. So this type of, it's a very simple mechanism, very simple tool. So this helped in making the complex laws into like in practically possible. And there is a compliance in monitoring and awareness generation. So this is from some of our workshops we have conducted. So the key takeaways are we have a stronger legislation in India now, and we have included the CITES listed species into this, our Wildlife Protection Act in India. This visual knowledge tool is readily scalable in any language. And this is a very well participatory approach in which, with the help of the fishers as well as the officials, we can do this in a better way. Thank you.
Okay. Thank you so much, Livne. Do we have any questions? No. Okay, thank you so much.
Thank you.
Thank you everyone for attending today. Really appreciate it. And to all of our speakers and presenters, good job. And hopefully next Sharks International, we have full room. Thank you so much.